Great.
All right. So this is Bisulfite QC Bio Collaborative Workshop number 6 for 2021 February. This is the DNA methylation analysis workshop. And can you guys hear me okay? You just want to use the chat window, I guess, for most feedback, I guess? Yeah, okay, great. So I'm Sean Kokas. I've been doing bioinformatics for almost 20 years now, I guess, all at UCLA. And so this is the first collaborative workshop I've taught though. So I'm a little unsure how the timing is going to go, how the interactivity is going to go, and so on. So today, this has been taught last time, I think is 3 2-hour segments. This time we've reserved three 2-hour segments before. Now we're increasing it to 3 hours. Not sure we're going to use all the time or whatever. And we have a lot of hands-on components starting towards the end of today and then the next days. And the purpose of those is just to get you guys to actually get stuff working on Hoffman yourselves and so on. And so however long that takes, that'll take, because the point is to get you guys to successfully conduct some analyses. Okay, so the first part of this is going to be some more background and kind of theory stuff, and then we'll move to the application and with a kind of a worked example that we'll do together. You should have the slides from this morning. I emailed them out. Did you get those? You may want to open those on your own machine to follow along, and especially when we get to maybe the commands, maybe you'll want to have that as a reference, and maybe even copy and paste may be useful for that. So also, could you just send in chat whether you got the, you successfully got the email and got the slides? Yes, okay, good. All right. At the end of, for those that are taking it for credit, at the end of day 3, there will be a short 10-question quiz. I think I'll let you email me back the answers to that. And there's also a couple hours homework assignment to complete by the next week, like the beginning of next week. You'll have this coming weekend to do that. Okay. So yes, we're talking about DNA methylation here. So that is one of several possible covalent chemical modifications of particular nucleotides of particular instances, particular actual individual molecules. So it's a binary mark. It doesn't change the primary sequence, that being the ACGT. It can be inheritable, which makes it epigenetic, and that's a lot of the interest. It plays roles in regulation of gene expression and roles in development. It controls some transposon expression. It's a generally repressive mark when present. It's correlated with diseases, cancer, aging, and so on. So now that we can kind of measure it better than we ever could before with high-throughput sequencing, this has really become an industry of study. For the workshop, we're going to focus basically only on eukaryotes, where the methylation's on cytosines. And we're only going to focus on 5-methylcytosine, the most commonly studied, abbreviated 5MC. And so our main goal here is to be able to assay the difference between 5MC and regular C at either all cytosines in the genome or at targeted subsets or enriched subsets of such sites. So we have there's methyltransferases that put the mark on, and you just think of them as little decorations. You have a particular cell. It's got chromosomes, probably diploid in most organisms. Each of the strands in each haplotype has some number of Cs, and each individual C is either methylated or not at any one moment in time. And that's what we need to sort of be able to investigate these marks to measure them. Okay, So yeah, so this tends to be, there tends to be characteristic patterns across cell types and tissues in developmental stages. And the methylation state is also dynamic and responsive to external stimuli. It's not nearly as fast as, say, gene expression is. The time scale is more like days or weeks or years, but it is dynamic. Yeah, so there's these other flavors that some groups try to study: hydroxymethylcytosine, formylcytosine, carboxycytosine. They all are vary based on what particular group you hang off the cytosine. These are, we're not going to talk about these. The most common assays used to determine whether a given cytosine is methylated or not is the bisulfite conversion assays. And these see hydroxymethylcytosine basically the same as methylcytosine. And the other two they see as equivalent to a regular cytosine. Okay, so much is known about this, and there's much obviously still to learn about mechanisms and functional significance. The most important thing to know about any particular cytosine is probably the primary sequence nucleotide context in which that C appears. In particular, the immediately downstream 1 or 2 nucleotides is very important and is related to which mechanism is operating. So the main division is whether that the C of interest is followed immediately by a G, or if it's not a G, whether the next base is a G, or whether it's neither of those two cases. So the IUPAC ambiguous nucleotide code for anything that isn't a G is H. Most of the IUPAC nucleotides, not cases are like that. They take the letter and move one position in the alphabet if you need a way to sort of remember those. So people talk about CG context, CHG context, CHH context. The underlying letter there, the first C, is the one that is being interrogated for the status of the methyl cytosine mark. And the other bases are giving you the primary nucleotide sequence context in which this C appears in the genome. And you just give a hint here that yes, they are related to different mechanisms. This is these graphs here are from Arabidopsis. And we have various genetic knockout mutants and different methylation levels of genes and so on are different in those different mutants, and the patterns are different in those contexts. So you know that these various genes we're knocking out are playing roles in the methylation of those specific contexts. So down here in the corner with our cartoon, we've colored the context in our little cartoon down there in the bottom right with the ones that by context. So in mammals, of course, CG is the one mainly of interest. That's the green one. And the big thing about that one is that it's symmetric, meaning that if you have a CG dinucleotide in one strand, immediately on the opposite complementary strand, you've also got a CG. And the biology does tend to maintain those together. So they're either both, they tend to either be, very strongly tend to be either both methylated or both unmethylated. The CHG can do that too, because it's also symmetric in a way. There's an intervening nucleotide, but there is another C, two nucleotides away on the other strand. And the CHH is often called asymmetric methylation because the other strand doesn't particularly have a C immediately in the vicinity. For plants, all three contexts are important. For animals, as I said, it's pretty much CG. CG sites in the genome in animals are underrepresented. They tend to cluster at these things called CG islands, which are generally upstream of genes and promoters. And for genes that are active, those promoters will be unmethylated because this is a suppressive mark. In plants, we have, and also in animals, you have this gene body methylation where expressed genes that aren't too short will tend to have the CG mark in the middle. Plants do the CG body methylation as well, but they also place CG on the transposable elements and their heterochromatin, where T's and heterochromatin also get the CHG and CHH. Plants tend to have very transposon-y genomes, and this is one of the ways they deal with that. And for especially things like CHH, the subcontext, as it's called, is also important. That is what those particular HH nucleotides are. CAA, CAC, and so on. If anyone has any questions at any time, just, I don't mind if you want to interrupt with through the microphone or the chat window is good. I've got the chat window popped up here. So in terms of the whole genome overview, I'll get the scale of an entire chromosome. Here's what a typical plant chromosome looks like. This is, again, Arabidopsis. We're looking at chromosome 1, where the centromere is in the middle, and Arabidopsis has its heterochromatin packed around its centromeres with fairly good boundaries between where that starts and stops. And the yellow is the repeat content, just sequence similar, dispersed repeat sequence similar. That's not any kind of methylation assay. That's just in silico investigation of the sequence, if you want. The upper right panel there has got the red, blue, and the green, blue, and red, which is the CG, CHG, and CHH methylation levels. So you can see that it tends to place the mark in that pericentric heterochromatin. And generally, always CG is the one that tends to vary the most from kind of near 0 to near 100%. It tends to be more binary on or off. The CHG tends to be more intermediate, and the CHH, when it's is generally very low, and when it's there, it's not necessarily at 100%. It's more modest in its variation. So the variation is, you should think of as relative to the extremes that it takes. So that CHH level looks low there, but in terms of the genome and individual places, that's as high as it gets, and that's a high CHH place at the centromere. You see the gene body methylation CG in the bottom left corner there, and you also see the various TE elements and so on get all 3 marks. In animals, your typical local landscapes looks something more like this. You have CPG islands where the sites are clustered. Those are often upstream of a gene in the promoter region, and they'll be unmethylated for active gene. In the intergenic space, you'll have methylated CGs. And at TEs, you'll have methylated CGs. And for the workshop, to keep things simple, we're mostly going to focus on human and mouse. Those are generally of the most interest, and the CGs are usually probably the ones that are more often functionally associated with things. As I mentioned that aging is correlated with methylation state. So as you get older, you tend to become globally hypomethylated. And that enables people to do this sort of DNA age clocking, where you can measure methylomes of people of a high diversity of ages, and then find sites that sort of correlate with their ages. The methylation state, of those sites correlates with age. So you have some sort of model that takes the methylome and produces a predicted age, and you run that on new methylomes. And so you can sort of then compare someone's physical age, their number of years since they were born, versus sort of their methylation state age, which might vary in disease conditions or so on. You can also do things like cell type deconvolution, where since the methylation pattern is sort of characteristic of certain cell types, you can collect reference methylomes from different cell types, and then collect some sample of unknown mixture, use the methylation samples to sort of figure out what fraction of the sample is which cells, which, break down the unknown mixture into proportions of which cells you think it is based on the methylation profiles, and then even possibly deconvolute expression profiles, sort of try to undo the mixture that way, once you know what's been mixed. Okay, so in human, let's just see how big of a problem we've got here. So the current human genome GRC H38, which is also the one Ensemble Release 103 current Ensemble release uses, the human primary assembly, that is chromosomes 1 to 22, X, Y, the mitochondria, 179 unplaced scaffolds, the ones that are still left, which are given as haploid nominal plus strands, right? That's the way it's distributed. That's about 3 billion bases. It depends on whether you count gaps or not. The ends, capital and lowercase n's that are placeholders for sequence not known. Of course, if you consider 2 strands, which we sort of need to do for DNA methylation, since it's a C, property of the Cs, and it's not a base pair thing, it's a base thing. So we should really think of the genome more as 6 billion bases for the purposes of methylation. And the human genome is about 41% CG content. And so the CPGs, if just based on one base statistics, should be roughly the square of 0.4. That's about 16, 17%. But when you actually go and count how many sites there are in the genome, there's only about 60 million. So it's only about 1%, which is 17 times lower than sort of the the CG content you'd expect it to be. And one of the reasons for that is because 5-methylcytosine tends to mutate very rapidly because it deaminates and it becomes a T. The C becomes a T under deamination, and the methylated C becomes a T under deamination. And that TG gets resolved by mechanisms to become really a TG is not a correct base pairing, and so it gets resolved. So, it can be resolved as a TA, fixing kind of the other strands to be the correct base pair for the T becomes a TA. So then you've mutated the sequence at that point, versus an unmethylated regular C, which deaminates to a uracil, a U, which also isn't part of DNA, but base excision repair knows how to put that back to a C, and so it sort of doesn't. It's not like the other path. And so the C to T at methylated Cs is like 10x the rate compared to regular Cs. And so some of the Cs tend to disappear, especially in mammals where the generic, the majority of CPGs, the majority of Cs in CG context will be methylated. So that's why they tend to go away. Now you may hear people say, oh, human has 30 million sites. Well, why? I said 60 million up above. Well, it depends on what you call a site, right? So, for CHH it's less confusing, but for CG, where they come symmetrically unpaired on 2 strands, people vary whether they call the pair, the CPG pair on the 2 strands 1 site, or is that two sites? I will generally call it 2 sites. It's then uniform with CHH and so on. It's really more of a confusion that only arises with CG. There are way more HGs and CHHs. 254 million CHGs, 890 million CHHs, right, compared to the 60 million CGs. Okay. Now, when we go to do, compare tissues or compare disease conditions or whatnot, we're interested in variation of the methylation. And so there's different things we might identify in doing that. So at the most basic would be a methylation variable position. That is a single site whose methylation varies across whatever you're studying. So that's an MVP. Then we have DMRs, which are probably the most commonly thing that people are concerned with. And that's because generally individual MVPs can be, they're basically hard for us to measure in a cost-effective way, at least at the whole genome scale, and they can be biologically noisy. So one way we handle that is by passing to little intervals of them, especially since, say, CPG islands are in gene promoters and they pack a bunch of CPGs in a row, so you get to observe a whole bunch of them in a little localized area. So DMRs, differentially methylated regions, are generally small genomic intervals, like a couple hundred bases, containing sites of correlated methylation level. Sites always means cytosine sites. And the region as a whole, since it's correlated, you can talk about the level of the region, sort of, and mean or whatever. And the region as a whole tends to vary over the thing you're studying. And then people have, they break into subtypes. There's IDMRs for whether the variation is due to imprinting, or TDMRs from tissue, or RDMRs from reprogramming, or CDMRs from cancer, or ADMRs from aging, and so on. Then there's variably methylated regions, which are like DMRs, but it's not the mean level that changes in the region, it's the variability of the region. It's more like the variance, It's not you're not testing the mean to change, you're testing the variance or standard deviation to change. There's allele-specific methylation, where since we're diploid organisms, You could talk about, there are cases where the methylation state depends on the parent of origin. And there's haplotype-specific methylation where a whole haplotype, meaning co-inherited SNPs or variants in a region, a haplotype, an inherited block, controls some methylation. In the workshop, we're gonna focus on pretty much DMRs, and not even MVPs much, although the tools that compute DMRs sort of have to deal with it. But we're gonna focus on DMRs. Okay, so how are we gonna measure these things? Well, basically, the options, including before the current situation, look pretty much like this. The basic plan was to turn the problem of measuring possibly methylated, possibly not methylated DNA into some problem that doesn't depend on knowing that difference, and then identify transform the DNA in some way that's sensitive to the methylation state, and then identify the transformed DNA. Because we have so many technologies for identifying DNA, just ACGT stuff. So the three main things that people used to do the transformation were things like methylation sensitive restriction enzymes. So you do a digest and either it cuts or not, depending on whether the site's methylated or not. People do immunoprecipitation, where you have some antibody that's sensitive to methylation state. And then people do sodium bisulfite conversion, which changes regular unmethylated cytosines into uracils, which can then become thymines once you do PCR. And so these are our three main ways of changing the DNA way that turns the rather difficult to deal with methylation mark into something of a more, something we can attack with just regular ACGT methods. So then once you transform the DNA, you have various methods you might try to examine the DNA. You might do microarray hybridization, that includes tiling arrays. You might do low throughput standard sequencing if you're interested in one locus. You might do high throughput modern sequencing if you're interested in a lot of loci. You've got qPCR, you've got emerging technologies like Oxford Nanopore that can actually bypass the three methods I talked about changing the DNA and actually have some sensitivity directly to the modified nucleotide. So a lot of these combinations have been tried, and they all have advantages and disadvantages. But the current modern standard is to combine the sodium bisulfite conversion with high throughput sequencing, mainly the Illumina platform, the current instantiation of which would be the NovaSeq platform. And so the main assays that people do of that type are WGBS, whole genome bisulfite sequencing, the original flavor. I'm actually the first author on the Nature paper that introduced this we did it in Arabidopsis, because the sequencers were so much lower throughput then that besides us personally being interested in Arabidopsis, it was a much more accessible genome at the time. It's still, though, high cost for large genomes such as human. And that motivates things like RRBS, reduced representation bisulfite sequencing, where we pass to a random subset. Hopefully, you use a restriction enzyme and then some kind of a selection process, on the wet bench, to try to enrich for regions that are more likely to be interesting, CPT islands and so on. And because it's sort of a sloppy enrichment, sample to sample, you're not going to get quite exactly the same regions and so on. But the big advantage is it allows you to concentrate your sequencing energy on smaller subset of the genome, and so you get better depth of coverage on parts. But you've made a trade-off there. You're not investigating the whole genome anymore, and you don't really have fine control over which regions you're getting, and it's a little hard to get consistency sample to sample which regions you get. So that leads to the next thing, which is targeted bisulfite sequencing, where you capture designed regions. That's an emerging technology that tries to lower the negatives of RRBS while retaining some of the positives. And if cost wasn't an issue, we'd all be doing WGBS for everything, but it still is an issue. So in the workshop, we're not going to really cover single cell, which is, I didn't even mention there, or the non-high throughput sequencing based methods, and we're going to focus on WGBS. the rest are really specializations of it. They do have analysis issues that are important and interesting, so on, but there's a limited amount of time we have in 3 days. So we're going to focus on WGBS. Okay, so how does the sodium bisulfite work? Well, just like in the natural DNA, we sort of have this deamination thing that can happen. That's what sodium bisulfite does. In this case, it deaminates unmethylated cytosines and turns them into uracils. And then PCR will turn the uracils into Ts. The methylated cytosines are resistant to the bisulfite. This is a fairly violent process. In particular, it breaks the base complementation because you have Cs, which are paired with Gs, and then some of those Cs turn into Ts, and T doesn't pair with G. So this is sort of why you end up with 4 strands if you amplify bisulfite converted DNA, and the original DNA doesn't base pair with itself anymore. And it's, as I say, it's violent, so it tends to take large, stable, double-stranded DNA molecules and turn them into small fragments of single-stranded DNA with wacky base distribution. Because in humans, say, if most CpGs are you have a region where you have unmethylated Cs, like a CpG island or something. Well, all those Cs just turn into Ts. And so the CG content, the ACGT distribution of your what eventually become your reads, the Cs all went away and turned into Ts, so there's almost no Cs left. And so the base composition is completely skewed. The T and A level, the T level isn't the same as the A level. The G level is higher than the G level is about where it was, but the T level is higher than the A level, and the C level has gone almost to 0. And that creates a lot of issues for PCR, because PCR has sort of exponential biases as base composition goes wonky, and the sequencers don't like it either. Their image analysis, auto calibration of image analysis parameters doesn't particularly like it, and so on. So there's all kinds of issues with the converted DNA, but basically this is a great way to adapt the current high but sequencing platforms, Illumina, namely to become sensitive to the methylation state of cytosines or not, and that's why we do it. So there's this good Zymo page if you want to get some, they've got a good write-up for some of the issues. So we're going to illustrate this process here just because this is important to understand how the assay works. So here's the starting state. We have a double-stranded DNA with some sites methylated, some sites not at the cytosines. And I put in the important ones to watch are the unmethylated ones. And so that's the ones that got these yellow bubbles on them. So the first thing to do is to melt the strands. So I did nothing besides just push the strands apart here. And then we apply the bisulfite treatment. And so those guys become uracils. Then we PCR. So those gives us A's against those. And further rounds of PCR to get back to the, flipping back and forth between those strands, we'll put a T at those places. And one of the things the modern bisulfite library prep protocols can do is they keep track of which strand was the original strand. And so even though we've kind of got 4 strands here relative to the original, they know which was the original, and we can arrange it so that the first, Illumina has this paired end stuff where you have a nominally double-sided, kind of effectively double-sided insert, although it really only exists in 1 strand at a time, and you only sequence 1 strand at a time. But you kind of can do paired ends where you can read, this end of the one and this end of the other. But there's, if you're doing single end sequencing, you only get this one. But anyway. So the first end that you sequence, we can arrange to be equivalent to the original strand, that equivalent to a bisulfite conversion of the original strands. We don't have to deal with this question of the 4 strands post bisulfite conversion. So these are the so-called directional BS library preparation protocols, and all the modern protocols are of this type. And so I'm just going to push the 2, this is our final kind of product that we're going to sequence, at least what we're going to see in the first end sequenced on an Illumina platform. I'm going to push these 2 back together now and bring in the original for comparison. And so what we've done by doing this is to effectively turn all the unmethylated Cs to T. And if the sequence fragments are long enough, 50 base pairs, 100 base pairs, you know, whatever. I mean, these days probably 100 is what people mostly run. 50, you know, 50 is okay, but depends on the genome. But if your reads are sufficiently long relative to your reference genome, you can still generally figure out from the bottom picture where they must have come from on the original genome, even though some of the bases are been changed now. But you're now expecting any any of the C's to change to T's, and you can make alignment with allowance for that. And assuming your fragments are long enough, then you can still figure out where they go, even uniquely. And so that allows us to effectively recover the methylation state of all of the original C's in the original strand that we started with. For any given read length, there's going to be some fraction of C, cytosines that aren't accessible due to ambiguity in the alignment and so on. This can be one reason to cut all of your samples if they vary to the same read length. We'll talk about that later. Now, effectively, the base complexity, the information entropy from the reads is a little lower than it used to be because we've sort of sucked up some of the entropy to use to distinguish the two cytosine states, which we can't use for alignment because we don't want to preferentially align certain methylation states over others. And so for the purposes of determining the location of the reads, we've used some of the entropy and information. And so the reads have to be a little longer than they would have to be otherwise. But, in the GA1 days, up to GA1, GA2, we were doing, 36 bases and aligning them to human and so on, RNA-seq and so whatever. It was more, it wasn't as good as it is today, but it wasn't terrible. I mean, it's, these days 50 is the shortest people run, and it's even more common around 100 bases. It's not really, the alignment placement issue isn't really that bad. Now, as I said, in mammals, for example, the global %C after this conversion, if you kind of just take randomly fragment the whole genome, apply this conversion and so on, you're going to have the C percentage in your library be extremely low, near 0. The T percentage high and not equal to A. So that drives PCR crazy. It drives the Illumina base caller crazy and so on. You tend to get highly uneven coverage because certain places like to amplify better now. You generally need a brightener spike in for Illumina. That would often be the Phi-X genome, which is roughly ACGT balanced, maybe even at 30% level. If you're running a lane that's full of bisulfite stuff, you may be putting in 30% Phi-X to get that ACGT distribution back into normal and let the base caller and image analysis sort of operate in a regime they were designed to. They don't want, anyway, otherwise your base call accuracies can be really, really off. Short fragments can be a problem. As I said, bisulfite conversion is pretty violent. The fragments that you get out of a BS library prep protocol can be pretty short, really short, if things don't go well. That means that longer read lengths may not actually help you, because if your reads are if your inserts are so short that your sequencing platform is trying to read beyond them, you're essentially going to read into the adapter that's put on those guys, and that's no good at all in terms of the adapter doesn't align to genomic sequence. You wouldn't want to consider any C's or T's in the adapter read to fake you out for crediting methylation or not to individual sites. Anyway, it could be a mess. So yeah, we're gonna have to make sure we trim away adapters, and if all your fragments are so short, you're reading into adapters with a given length. You're doing single and 100 base sequencing, but your inserts are all 50 bases. Well, that back half of the 50 isn't any good for you, and you're not getting your money's worth. So. And for that reason, also paired end, maybe this is one of the reasons why paired end isn't often done. It's because if you want to do paired end, well, 50-50, maybe that's the same as 100. But paired end 100-100 to get 200 bases, well, for that to work, you better have inserts that are 200 bases or bigger, because if your inserts are shorter, then you're starting to double sequence the part in the middle. Well, at least the aligners recognize that these days, and they won't double count the stuff in the middle, but you're wasting your throughput at that point. If you get to the point where they totally overlap, you might as well have just done single end sequencing. And beyond that point, you're even reading into adapters on both ends, right? So to make PE worthwhile, you got to have inserts long enough to make it worthwhile. And also a little point also is that on the Illumina platform, at least typically the protocols that generate paired ends, if you do sequence the second end, that usually comes from effectively the reverse complement of the first end, which is one of these other two strands post-PCR in bisulfite. So rather, if you call these strands sort of the C-poor strands, people often say, or the T, maybe T-rich strands, or whatever you want to call them, the ones from your second main end are the G-pores and A-rich. It's almost like you did bisulfite that converted G to A, because you're looking at the reverse complement of a bisulfite converted product. And so don't be surprised when your second ends have a completely different ACGT decomposition than your first ends. That's completely expected, and biosulfite aligners that are pair-end aware will already know that. They know to switch to a mode where it's effectively a G to A conversion. Another problem is under or over conversion, mostly under conversion. So that is where secondary structure or whatever it is, we don't entirely know, inhibits, maybe the strands don't melt or whatever. The sodium bisulfite can't get in there. You get little bubbles if you have a 100 base read or something. There may be a little bubble in there of some dozens of bases or something that didn't get converted, right? Where the unmethylated cytosines in that bubble didn't get the bisulfite treatment, and so they stayed Cs, and the assay will think that they're methylated Cs because they're, that's what it looks like. So that can be a delicate issue. It's not as much of an issue for CG and mammals. It's more of an issue for plants, where CHH is low and interesting, and sort of having non-conversion at even low rates can be comparable to the signal of CHH, the biological signal of CHH. And so handling that is quite delicate. I can't really get into that here, and various pipelines or aligners may or may not have support for that. The support may not be appropriate for the regime you're operating in based on the statistics of methylation in your genome of interest, especially if you're working in a non-model organism. And so it's something you may have to be aware of, but it's kind of, we can't get into it here. If you've got library prep questions, Suha in Jacobson lab and Marco in the Pellegrini lab. Marco has a library prep workshop. They are real library prep experts. Some people actually spike in completely unmethylated and completely methylated DNA as controls, but it's sort of difficult to make those really great perfect controls. Leave it at that. If you want to see what non-conversion looks like, here's an example from Arabidopsis, where here we're looking at a close-in view of some regions. The red boxes would be apparently methylated DNA based on bisulfite reads. The little reads here are bisulfite reads. The red marks things where the read declares. The read makes it look like there's methylation there. And what you see is that the methylation is not kind of vertical, it's horizontal, meaning that the methylation is all coming from a subset of the reads. So it's like certain reads are like all methylated or not, and it's not kind of distributed randomly across the reads. So even though the level is maybe 50% or something nominally by looking at this picture at some of these sites, that's all coming from 50% of the reads and not a different 50% as you look site to site. And that's kind of a flag that maybe this is going on. So if you're kind of looking at alignments in detail and you see something like this, you might start to wonder if you've got non-conversion. Okay, and another big issue is duplicates. So, PCR has been driven crazy by the base composition and so on. One of the problems you can get is low complexity libraries where a small subpopulation of genomic sites sort of took over. And so that may mean that when you put them on the sequencer, you're not really getting sites all over the genome. You're getting a couple sites over and over. Well, not a couple, but a subset. And that's not good because those are sort of duplicates that arose through artifacts of PCR. And those multiple observations at a given locus then don't really represent independent queries of real cytosine molecules, real DNA molecules from the original biomaterial. So that's not good. The sequencer, that often happens during the library prep stage because bisulfite often needs quite a few PCR cycles. Bisulfite libraries often need quite a few PCR cycles to kind of recover from the treatment. And so that plus it's sort of everything's working against you in terms of biases for PCR with this kind of stuff. The sequencer itself can also introduce problems. The so-called optical duplicates or XAMP duplicates on the instrument, because we have to do a little bit of amplification on the instrument to measure these prepared constructs. The best solution is UMI, unique molecular identifier, a modern technology, and so on. And not all protocols can support it or have been adapted to it, but when you can do it, it's a good idea because it's sort of the cleanest and most complete and easiest and on the computer to do, take it out once you've done it. It's the right thing to do, and you can do it. Otherwise, you're up to things where like you kind of, if you have paired ends, because the insert length is supposed to vary a little bit, if you have a whole bunch of reads at a given locus that all start and end in exactly the same place with an effective insert length that's exactly the same, tend to suspect that's PCR duplicates, because with the insert length variation, if they were randomly selected, the chance that they all really came out exactly the same is low, and then you might just keep only one copy. With single end, you don't really have that PE length variation to help disambiguate the cases. And now you're down to just sort of picking guys that just pile up at the same place. Depending on your coverage and a whole bunch of other things, whether that's a good idea to filter out things that look like duplicates that way or not, it kind of varies. Now for certain assays like RRBS, single-ended RRBS or something, you really can't filter them out at all because the passage to restriction enzyme selected locations means you are expecting a huge number of guys to start in the same place at the restriction site. So this is also a delicate topic, but a lot of libraries do come out with a lot of dupe problems, and so it's something you often need to do. But it's kind of a delicate topic and depends on details, and I can't really get into it here. If you're looking at a bunch of libraries like this, here there's 4. The one that looks most like it probably suffers from a dupe problem here is the bottom one, because if you look, the rest kind of jitter around and so on, whereas this guy tends to all be these vertical stacks, calling the same methylation state with exactly the same start and ends. This is the one you would suspect most of being in trouble. So I mean that's kind of what it would look like as a if you see that, you start to think about it. And then all these issues that we just talked about can be differential across the samples being compared, which can be an issue. And this also makes doing meta studies across different studies, reanalyzing data from multiple studies, also problematic, especially if you don't do some kind of uniform in silico reanalysis or something. Methods have varied a lot in the past, and they still vary today a lot, and so on. Anyway, it's things to worry about. Okay, generally here in the workshop, I'm trying to give you, not really absolute best practices, but I'm trying to just educate you to a whole bunch of issues so that when you conduct your own actual research, you can make intelligent decisions, right? You can evaluate tools, you can read methods and review papers, you can intelligently kind of pick something that's going to work for you, and you at least have some awareness of things you might need to be worried about. There's all kinds of tools for different stages here. We can't really get into them. There's hundreds of them at this point of both tools and papers on this stuff, and I can't really cover it and so on. And even if I tried to make some sort of best practices this moment, it'll change by next year or something. So anyway, that's sort of the goal of the workshop, is to really be more educational and awareness and give you some good stuff to do, but not necessarily the best or something that's most appropriate to your particular situation. Okay, so these particular these 3 assays I mentioned, the main ones here, WGBS, RRBS, and targeted, work something like this. You get your input material. In WGBS, you put the adapters on first. You bisulfite convert. The adapters are pre-methylated, so they don't get mutated by the bisulfite conversion. And then you amplify and go on to the instrument. With RRBS, you do digestion first with a restriction enzyme. MSP1 is a good one people use a lot. Then you do the adapters, then you do bisulfite conversion, and you're back to where WGBS was. For targeted, you do bisulfite conversion early. Then you got to pull them down somehow. Maybe you got design probes and so on. I mean, there's different ways. This is an evolving area. Anyway, obviously, when you actually get into it, the actual steps of the individual protocols vary, and there's multiple protocols for each of those generic types of BS-Seq. Yeah, single cell assays are currently rare. Most assays are randomly pulling fragments from huge gamishes of lice cells. So each cytosine in a particular haplotype of a particular cell is either methylated or not, binary, 0, 1, 100%, 0 or 100%. So the assays are usually averaging haplotypes in cells, generally cells of mixed type and stage. And so the result is that each C site on the reference genome, the two-stranded haploid reference genome, has essentially a continuous methylation fraction, anywhere from 0%, all copies, all haplotypes, all cells in the mixture are unmethylated, to 100%, all copies are methylated. But the stuff in between could be all kinds of different combinations. If you measure a 50% level of methylation, that could be all cells have one haplotype methylated and the other not. 50% of cells have both haplotypes methylated and the other 50% have neither. Some, different fractions and splits among different subpopulations or whatever, you don't know. All you know is that the average across all the molecules in your sample, hopefully the assay is sampled those more or less uniformly without bias, is 50%, right? That's all we know. But sometimes the interpretation of that is tricky. And sometimes people do tweaks to the assays to try to limit it to particular things, particular haplotypes, whatever. I mean, whatever you got to do, you got to try to, if you're concerned about those details, you got to find some way to detangle it. Okay, so when we look then at the reads we get that cover a particular locus, a particular cytosine site, unfortunately, given the current state of technology, the pileup coverage on individual sites is usually modest to poor. So low count, small sample size n statistics and random sampling come into play when you independently try to estimate a single sample's percent methylation at each site. It's like RNA-seq if all your genes were, like 10 counts or something. I mean, this is kind of the region we're down here in this low counts zone. So each read, let's look at this top strand example here. Each of these, we've got 1, 2, 3, 4, 5, 6 reads here that we've BS aligned to this place that cover this place. And we're only concerned about this one particular C right now. So these are the six reads that we kind of shoot the spear up from the reference genome there, and these are the 6 reads we skewer. Each of these reads is from a random haplotype of a random cell in the original biomaterial. Right? So there's, how many total haplotypes and cells are in the original biomaterial? A lot, right? Not quite Avogadro's number, but big, right? So effectively, it's a continuous. The denominator, the true denominator is gigantic. We have no hope of getting anywhere close to the true denominator. So we're going to try to estimate the methylation fraction of this site in the original sample from just these 6 reads, right? That's all we can do with our current technology. So our best estimate, the MLE, most likely estimator, is basically the obvious thing, right? You're going to basically take the number of methylated observations the reads declare over the number of methylated with unmethylated observations the reads declare. What else could you do, right? I mean, obviously, that's your best guess. So here, translating that into the language of bisulfite conversion, that's the number of observed reads with C's over this place over the number of places with C's or T's, right? That's the meth over meth effectively. And here that's 2 over 2 plus 4, right? We see 2 C's, 2 reads with C's, and 4 reads with T's. The coverage here is 6. So that would be 2/6 or 1/3. And our best guess of the fraction methylation at the site in the original material is 1/3. Okay. In the bottom, strand example here, what would be the best guess of methylation here? Well, this one is a little more, interesting in a way, interesting. So this one's got some things that look like either base call errors or could be genetic variants. We've got a gap in the alignment here at this base. We've got a low quality base call C. We've got a regular good quality base call C. We've got an A, and we got a regular good quality base called C. Well, we probably throw, the deletion. What do we do? No observation, right? Whatever. The low base call C, it's probably not a good idea to take the base call of things that the immuner base caller isn't very confident about. If the base caller thinks, well, I've got a 30% chance of being wrong, maybe we're not going to take that observation, because we don't have many observations of quality, right? We don't want to contaminate it with, with a third being junky. So, and we're gonna, that A, well, what can we do? It's either a base call error or a genetic variant. It's certainly not something we can be consistent with the bisulfite tree. So out that goes. And effectively our coverage here is 2. They both called methylated, and our best guess for the methylation fraction here is 100%. Obviously, some genetic variants could appear to induce what look like methylation or unmethylation calls, and that it's not generally a problem. It can be a problem. Some alignments may even get fancy and start to incorporate DB-SNP into the analysis and so on, whatever. But anyway, how much of a problem that is for you sort of depends on the details. Sometimes that's what helps you, because sometimes that's how you attack the haplotype problem. The haplotype specific methylation. But anyway, you can obviously see here that if coverage is low, you can't really be too certain about the methylation in the original population, the average methylation in the original population at that site. You just get unlucky, If the coverage is 1 or 2, you're very likely to get 0 of 2 and declare 0% methylation, or 2 of 2 and declare 100% methylation, even if the true percent is somewhere in the middle, right? As I say, it's kind of like RNA-seq, where all your genes are kind of like at the bottom end of the expression spectrum, where you usually throw away those guys. You don't even start to statistically analyze genes until they're up into the dozens to hundreds, and you've got genes in the thousands of counts, even tens of thousands of counts, even hundreds of thousands of counts in a typical RNA-seq assay. We don't have anything like that here. To give you an idea of what you can get on a current platform, so if you take sort of the throughput, winner right now, which would be a Illumina NovaSeq 6000 running an S4 flow cell. One lane of that, maybe you get 80% of the actual constructed nanowells, the lithography constructed nanowells on that thing get occupied singly by your constructs. That's the PF1, maybe 80% PF1. That's actually a pretty good number. And then maybe you got to spike in 30% Phi-X though, so you don't get all of those 80% wells. 30% of them are going to go to Phi-X as your brightener. Or maybe you should throw in at least some non-WGBS libraries of interest, so you can actually get some useful sequencing out of that 30%. But anyway, that'll get you about 2.1 billion reads. If those are single and 100 reads, or maybe paired in 50-50, I mean, we'd like to do paired in 100, and that's 200 bases, but like I said, the fragment lengths are usually a problem with BS libraries. So let's say you're doing single and 100 or paired in 50-50. That'll give you 200, just multiply by 100, you'll get 210 billion nucleotides out of that. And with the human genome size, that's about 36x coverage of the bi-stranded haploid human genome. That's not bad. That looks pretty good to me these days. When we started on GA1 with the Arabidopsis, which is a 25x smaller genome, we had to run almost 100 lanes to get our, and then 30 of them didn't work or something, to get our average 10x coverage that we did for the Nature paper. So it's it's orders of magnitude improvement, but but the genomes are big, and people, you want to run bioreplicates, you want to run lots of samples to compare and so on, and that 36x coverage from a relatively expensive lane, that's a many $1,000s run there. That's not a lot of samples to very high def. Even at one sample, there's some views in which you would say 36x coverage is not even that a really high def, even before you multiplex, if you're going to multiplex. But with the Arabidopsis, that same setup gives you 890x coverage, and now for sure you're multiplexing and so on, because that's important. But anyway, so ideally your statistics deals with this. But a lot of the stuff people do doesn't. And a lot of the time people just work with the point estimate, just the C over C plus T that we've been talking about so far, and they just take that point estimate and run with it. And hopefully at least you've imposed some kind of minimum coverage, that denominator C plus T, maybe you force it, maybe you only look at sites with at least 5 or something, I don't know. But some sites then are essentially unobserved, and which subset of sites it is tends to vary per sample. And that comes into it when you start comparing samples against each other, because you don't always have, observations on top of, not all samples can be observed at all sites. To give you an idea of what the fuzziness kind of looks like with those low denominators, one nice model for this would be to treat the fraction methylation as a Bernoulli weighted coin flip, and then then you get the binomial distribution in terms of observed. If you're going to observe 5 reads, then you get a binomial distribution with that weighted coin flip as the parameter in terms of how many reads you actually observe methylated, even though the true fraction is just the one thing. You're going to, of course, observe a variation around that based on the random sampling of your discrete reads. And if you put a beta distribution prior on that weighted coin flip probability, the fraction methylation, you get a setup that's known as a conjugate prior, in the sense that you start with a beta prior, you observe, 3 out of 5 reads to declare methylation. The posterior ends up being another beta. And the way the beta parameter is updated is extremely simple. If you started with a beta AB and you observe M methylated and U unmethylated reads, the update is just beta A plus M, B plus U. So you just kind of add the first to the first and the second to the second. It's very easy. And there's some very easy priors that you might start with. AB11 is a uniform prior. That's sort of reasonable. Another one is 1/2, 1/2. Kind of looks like pseudo-counts at that point. That's Jeffrey's non-informative prior. It actually emphasizes the 0 and 1 aspect of it, which may not even be that crazy if you're studying CG methylation, since those tend to be binary on or off anyway. Actually, it turns out that the prior doesn't really matter much. Even with 5 observations, the prior essentially doesn't matter hardly at all. The left and right picture here are with those 2 different priors, and you can see how different how much emphasis on the 0 and 1 side Jeffrey's does there. So the prior doesn't matter that much because it disappears quite rapidly with even really small amounts of data. But you can see here, though, that the fuzz in the posterior distributions is quite large. So if you observe 2 out of 5 reads to declare methylated, that would be this kind of green curve. I don't know, that's pretty wide in terms of the region that's roughly a modest probability, right? And maybe anything from 20% to 80% is plausible, right? So you haven't really pinned down the true value very much. It's because that 5 isn't really that great. So even if you bump it up to 50, which is, that's luxurious BSseq coverage. Yeah, you're living the high life. Even now, the methylation percentages aren't pinned down like you might want, right? It's not like you know them to 2%, right? Like you might think, right? Oh, I've measured 50 and the denominators, 21, 22, 23, they're all 2% apart, right? No. It's much fuzzier than that. Green now is 20 out of 50. And now if you look at this green bump, it's like, well, maybe 30 to 50% is plausible. Well, at least it's narrower than it was before, but it's not 2% wide. Right, and this is at a single site. And now if you want to start doing differential methylation, you got to compare 2 of these empirical things that you measure from different samples. So they're going to get a little wider as you kind of do the subtraction to kind of find the difference in methylation between the 2 sites. And now you can see that like doing differential methylation at individual sites with the kind of coverage that we typically have in today's experiments is not that great. This is a big reason why people do DMRs, where you get to pool the data among multiple sites, because then you get the denominators to build up from a couple sites. Right? You get a lot more statistical power and so on. But this is the facts of the situation we're in. It's like parts of RNA-seq experiments that people throw away. That's the entire experiment here. Okay, we're all down here at the left edge of this coverage, distribution RNA-seq. It's a little better for CGs because, like I say, CG context, the Cs tend to be either off or on, and plus you get the benefit of having the 2 strands maintained symmetrically, so you kind of get a 4-free double covered bump. But if your decision is sort of, is the methylation 10% versus 90%, you don't have to worry about the middle so much. It's much easier. So like now we're looking at 10 versus 90% as the 2 possibilities for the true fraction. With coverage 5, well, actually, it's pretty clear. 0, 1, or 2 looks like an off, and 3, 4, and 5 out of 5 looks like an on. And with 50, it's 50, you don't need 50, right? You see how everything from 15 to 35 isn't even plausible with 10% and 90%, right? So you don't need coverage 50 to make that decision. Coverage 5 is more like exact. So that helps. But anyway, this is basically the whole motivation for RRBS and targeting, because it allows us to take our fixed sequencing dollar and concentrate it on something less than the whole genome, because the statistics with low denominators is just sort of dismal. Okay, all right, maybe I'll stop there for a second, and we're about an hour in, and let's give people a break, maybe a chance to ask questions. And I've sort of lost my chat window. Ah, there it is. Okay, so does anybody have any questions? Pop in or or chat or anything?
Hi, Sean. I have a question. So my question is basically because I'm interested in this hemimethylation for strategy, and I guess from the bulk whole genome bisulfite sequencing is almost like very, very hard to tell the hemimethylation.
Yeah, you're not going to, it's not sensitive.
Yeah.
Bisulfite's not sensitive. So you have to do something else, right? So
like single cell, I guess.
Well, there's other protocols. So there are papers about this, and there are different treatment protocols to do it. But yeah, straight up bisulfite doesn't see the difference. So
gotcha. Okay, thanks.
Anything else? Okay, people following along. This is all stuff you already knew. I mean, hopefully I gave you some things to think about, right? I mean, if you already knew stuff, maybe there are some aspects of it you can appreciate now, and maybe you got to keep in mind when you do the analysis and so on. Okay, so now we're going to move on to the actual mechanics of how to conduct this kind of analysis. So let's get this going again. So this is what sort of a generic WGBS pipeline would look like. We're going to start with some Fastqc files, which are the individual reads. Generally, the sequencing cores these days will demultiplex for them if you demultiplex them with any sort of normal strategy. And so you'll get one set of files per sample. Well, each lane has its own set of files. So sometimes, I mean, these days, probably people are less likely to run an individual library in multiple lanes to build up coverage. That used to be very common because, the amount of coverage needed for a human-sized single sample WGBS until recently was generally beyond the ability of single lanes. So you didn't multiplex at all, and you needed multiple lanes. But these days, you're starting to get to the point where you would even start to multiplex even human samples multiple in one lane. You probably don't need to run one sample multiple lanes. But anyway, each lane would have its own set of files, and then within the lane, you have one or more samples. And because of the demux done by cores now, you will get one set of files for each sample within the lane. And then if you've done paired end, those are generally in separate files too. Main end 1, main end 2 will be in separate files. Obviously, when I say sample, different bioreplicates of the same condition are a different sample. Conditions are collections of samples, hopefully more than one in each condition, because biological noise and variation and so on, there's really no good way to get at that without having multiple biosamples, just like an RNA-seq. And although in RNA-seq, people get away with 2 or 3, and in bisulfite, people still do, 2 or 3. It's all because of expensive, right? I mean, it's this question of more biosamples or more condition. Obviously people want to err on the condition side. In the original Nature paper, we got away with no bioreplicates, but we're introducing the method for the first time, and the results from the mutants and so on are so clear, and so on, it wasn't a big deal. But yeah, you should have bioreplicates these days, 2 or 3, ideally 3. You probably can't afford more than that. It'd be nice if we could, because the variation is nice to see directly, right? Same thing in RNA-seq. If you had tons and tons of replicates per condition, you don't need any fancy statistics, right? You can do everything at a per site. For them, it's per gene. For us, it's per site, per site way. But otherwise, you have to start pooling across things and so on to get enough statistical power. But such as it is. So anyway, so that's the set of files you start with. With all those things that can go wrong in a bisulfite experiment, you're going to want to look and see whether those reads look good. And so there's some kind of quality control inspection that we'll do before we do anything. Then from the results of that, we will probably make some decisions. We may need to trim adapters because we read into them. There may be low quality, per base, low quality edges of reads. certain cycles may not look good, you know, especially in our RBS, say, you know, because you've selected with the restriction enzyme, you know, the beginning of those reads is going to be a little different and not representative of where we are, you know, kind of because of you synchronize kind of based on whatever context the cutter likes to cut at. And depending on the protocol, you may have some kind of sloppy ends, sticky ends or whatever, you know, overhangs. Those overhangs may have gotten filled in with free nucleotides, which won't be methylated, and you can't rely on the methylation information from those filled in points. So you may got to trim away a few edges, you know, bases on the edges. Anyway, we may pass to a subset of cycles because various cycles don't look good or aren't trustworthy. Some reads may be too short. Anyway, there's various things that we may do to clean up the reads before we'll continue with it. So there's a stage, this orange box is the stage for that stuff. After that, we'll probably do a second QC inspection to see did we take care of those issues. Then if you did UMIs, this is then a good point to do some dedupe. Otherwise, dedupe at this point is usually not done and sort of difficult because it's sort of an all versus all sequence comparison problem. Most people that do dedupe not via UMIs would do it after alignment. Then we're on to the green box here, which is the actual bisulfite alignment. That's very important. And then you're going to start your examinations some basic statistics, like, you know, how much aligned, how much are united uniquely, and so on. There's some sort of output file maintenance we got to do after that in the blue box. Then we'll probably start, you know, just doing some exploratory stuff and sanity checking. We'll look at the reads in a genome browser, like you've already seen some pictures of piled up reads and so on. We'll do stuff like that. At this point, we will, you know, you would do some deduplication post alignment if that's going to be something you're going to do. You would then reinspect in a genome browser to see if that problem's gone. And at that point, sort of the primary analysis is mostly done. I mean, in terms of we've got them aligned at that point. The next step is to pile them up and count the C's and T's above individual cytosines to sort of the bisulfite quantification step. If you like, sort of the equivalent of the quantification step in RNA-seq, where you sort of credit reads to gene models. Here we're crediting C's and T's to cytosines. Then once you have that, you basically got the primary analysis done. And now you start your downstream statistical analyses. And so that would be selection of sites that you're going to do differential analysis on, right? Because you got this issue of different sites have different covers, different sites aren't covered the same in all samples, and we have to pass to a subset of sites that we're going to consider well covered enough in enough samples to use. So we're going to choose our sites for analysis. We're going to just gather our data, collect our data, aggregate our data, reformat into some unified format that has the information we need at the selected subset of sites. And then we can run some statistical analysis to identify the DMRs. And at that point, we have a list of regions, and you can do functional annotation, what things are enriched near these regions, you know, genes or whatnot. You can do exploratory analyses, visualizations, plots, other downstream things, and so on. So, yeah, so today we're going to start on this process. We're going to do the initial QC, the trimming, work our way toward bisulfite alignment. I'm not sure we will get, I don't think we're actually going to get to actually doing the bisulfite alignment today. That will be the start of day 2. And then by the end of day 2, we'll probably be somewhere in the quantification stage, possibly into the DMR calling. And then day 3 will be more probably playing around with some of the annotation, exploring, visualization stuff, and whatever overflows. The tools, the particular tools that I'm going to use as examples, I'm not saying these are the best ones, but these are reasonable ones. We're going to use Fastqc for the QC steps. We're going to use cutadapt for the trim steps. We're going to use BS-Bolt from the Pellegrini lab, which is quite recent from them. And that's quite nice and does a decent job. That will be that actually integrates a lot of the steps, which is quite nice. So that will do the alignment, the quantification, and the aggregation for us. SAM tools will be our little alignment maintenance thing. IGV will be the genome browser we'll use. R will be R in various R packages. will be the statistical environment we will do some of the statistical analyses and visualizations in. DMR calling will be with Methylation, and then various things for the functional stuff, the web tools and whatnot. And the data that we will use hands-on here in the workshop is some human WGBS data from several years ago that I took from a Nature paper. And it's been cut down. I had to cut it down a lot to get, the file sizes would otherwise be gigantic and they would take too long and so on. So what I've done is cut them. I didn't, the paper goes through 40 samples or something. I cut it down to 6 and only chromosome 21. All right, so that's the smallest human autosome. And so that gets the data. I mean, the data is still even not trivially small, but it, that gets it down to something we can sort of manage online, hands-on during our 3 hours. So that's the plan. All right, so now now hopefully you can play along, right? So shrink this window or whatever, get a terminal window next to it on your local machine, and log into Hoffman, right? So I sent an email out on Monday, and hopefully you've all got your Hoffman account squared away, and you can log in. So that's with SSH. Maybe, yeah, I guess I will try to do this in parallel too, which means I've got to add another window to the Zoom screen share here. I don't know if I can actually change it. I just got to stop, I guess, and then reshare screen. And we do this guy. This shifts. Share. Okay. There are now 2 windows shared. So okay, so somebody, I lost my chat window. Where did that go? Okay, somebody tell me if you can see this terminal window. Is it too small? I mean, is it readable? Is it visible? Yes, it's fine. Okay, great. Okay, so hopefully you can open a terminal window too. Let me start from the beginning, just so I'm with you guys. So I'm gonna sign in. I don't know what your username is. Use your own. I have passwordless, crypto-based RSA login enabled, which is great and convenient. I can't really. I mean, we can go over that offline or something or at the end if you're interested, but that means I don't have to type my password. And it also means when I do file transfers and so on, I don't have to type my password. That's just convenient. Okay, so here we have signed in, and of course you all know that there's the login nodes where everybody using the system has to go through these login nodes, and we're all sharing this internet connection pipe into this thing, and this is our way we communicate with all the compute nodes. And so if people bog down the login nodes, it degrades everybody's experience. So we try not to do any real computation on these login nodes. So the first thing we do is we need an interactive session on one of the compute nodes, just that we can just use these login nodes as gateways and not really load them down. So we're going to, okay, into the next slide. This is complicated, managing all these windows here. Let me just shrink probably this a little bit. All right, hopefully you can follow along on you have the slides separately, so if things are covered or whatever, you've got the slides locally to look at as well. So QRSH, get a remote shell, and then we're going to ask for parameters, what sort of resources we want in our interactive environment here. So I want to have 4 cores on this whatever machine it gives me. So I'm going to say PE shared 4. I'm going to be able to have authorization to use up to 4 cores on that thing. And I want it to be interactive and give me a 6-hour time limit. When that 6 hours is up, the session's just going to die. Well, it doesn't matter. 5:59, 59, okay, 6:00:00 killed, right? This is a hard limit. And then I want 7 gigabytes of RAM allowed for us. And that means with our 4 cores, that's 7 times 4, we'll have 28 gigabytes RAM authorized for us. If we exceed that limit in certain ways, we'll also get our session killed. or individual things running in the session field. It's a little messy how this RAM limit works, but essentially we got 28 gigs of RAM and 4 cores. Okay, so I'm gonna, and hopefully that's not too crazy. If you ask for, the more resources you ask for, the harder it is to satisfy and the longer you're gonna wait. It's also possible to easily ask for resources that are impossible to satisfy in the system, and it doesn't tell you that it's impossible, and it will just wait forever. And anyway, so that happens. Well, I didn't have to wait very long. Okay, so I'm on node 7160, and somebody want to say in chat or whether they successfully got a session with those parameters too, or all right. Maybe someone else just to see that more than one person who's got there. Great. Okay. Okay. So now we're ready. This is going to be our little environment we're going to live in for 6 hours. Well, not 6 hours, but up to 6 hours. So the next thing to do is, there's different places to put stuff on Hoffman, different storage spaces. You got your home directory, which is where we're in right now. That's home, first letter of username. That's usually very tiny, right? But it's persistent and it's backed up, and it's where your most critical files go. There's the scratch space that's like uscratch cocos or uscratch whatever for you. That's fast flash memory that is where you do most of your work. You get 2 terabytes of quota in there, and it lasts for about 1 or 2 weeks. If files have old timestamps down in there, they get auto deleted with no warning, right? But you get 2 terabytes down there, right? And you may have some a third place you may be able to put stuff is some sort of space associated with whatever project that you got on when you got put on Hoffman, you got associated with some project, some sponsoring project. And that project may have paid for space, and then you can use that space too. But if it's like our space, it's always full. So the scratch space is sort of generally where you have to live with it, and just got to deal with this auto deletion issue and so on. And so hopefully you also have some local pile space on some personal machines, or you have some in-lab cluster of your own or something, and you have some place where you can kind of keep your permanent data and so on, and you sort of slush thing up to hop and scratch, you work on it for a while, and you slog it back and so on, and you kind of do this dance, and that's the way it goes. So for me, my scratch space is use scratch cocos. I've got some stuff in there right now. If you want to see your quotas and how much you're using of your various places, you can say my quota. You can see I got 3 places, right? I've got my scratch space. I'm actually using 870 gig right there. I'm almost half full. My quota is 2000, 2T. The units here would be in gigabytes. There's also a file limit quota, usually not a problem. But actually you can see you'll see in the Pellegrini space, we're actually using a substantial amount of our file quota. So each individual file, you have a limit on the number of file names you can also have. Usually not a problem. Another space here is my home directory. You can see it's very small, 20 gig, right, is the limit. I'm using 4. My project I'm associated with is project MCDB. I'm in the molecular cell development biology department. We have 13 terabytes of paid space, of which 12.5 is used, so not a lot free. And we're actually using 5.8 million files, and our quota is 13 million files. So just in file names, we're already 45% of the way to our quota on that. But of course, the space is usually the one that's relevant. Anyway, so you can type my quota, figure out where you've got space. You're probably going to be working in Scratch. I'm going to do this workshop assuming you're working in Scratch. So let's all, go to your Scratch space. Okay, so. Okay. So go to your Scratch space. So we're going to need to type that name a few times, so time to learn about shell variables. All right, we're going to give an abbreviation to this so we don't have to keep typing it. So we're going to make shell variable WRK be your Scratch space. So for me, that's this. Okay, that's now a shell variable, and now I can refer to it like this. You will see, some people would just write this. I will generally quote things and use the most conservative syntax, because once you've done shell scripting for a while and you've written enough scripts, you realize that all the disasters that can happen, and it just pays to be safe from the start. So those extra braces and quotes are there to sort of disambiguate certain cases that may not be relevant here in most situations, but there are edge cases where they're absolutely relevant and they'll bite you. And I will generally, to illustrate good shell kind of hygiene, I will generally use, even though it's more typing, I will use the sort of the proper escaping. Okay, so now you're gonna change to your scratch space. Okay, so now you're in your scratch space, and we're going to make a directory in there to work in during today. So why don't you make yourself a day one subdirectory down in there? Okay, so now I'm in my scratch space, and I have this day one subdirectory. Right? You're not going to have this other stuff, or maybe, I don't know, maybe you already got stuff in your scratch or whatever, but you got this day one space that we're going to do our work in today. Okay, and now what you need to do is you need to copy in the reads to use for the workshop. Okay, I've got the Fastqc files prepared in my scratch space, and you're going to grab them. So it's easiest if you change directory into where they are, and then we'll copy them by naming the path to where to put them. You could do it in either direction, but let's change into my scratch space, which has got the reads, so you can kind of look around there and see whatever. Now, the next thing to learn is tab completion, right? This is your friend, right? I like long descriptive names and so on, and the way you deal with it without going crazy, well, first of all, you get practice and you start typing 120 words a minute, but tab completion, right? So I'm going to just type this W, whatever, and hit tab, right? There's only one unique completion of that in that path, and the shell knows it, and so it types the rest of it for me. That's great. So there we go. I don't have to type most of it. All right, so I've got two subdirectories in there. There's a day one BS reads and scripts. Right now, the thing of interest is this BS reads change in there. All right, so this is the path we're in right now. And you should see 6 Fastqc files in there, okay? And now we're going to copy them into you make a copy for you in your personal scratch space. First, let's see how big these things are. That's 3 gig. So there's 3 gigabytes of files here. 6 files, 3 gigabytes total. All right, so let's copy them. CP, the V is verbose. That'll just let you know as it copies each one, kind of like a little progress bar. The I means interactive. If you already had a file at the destination with this name, it would let you know, do you want to overwrite it, yes or no, just a little safety thing. You're not going to have a copy, it doesn't matter. And then some people would just do a star here. Let me be a little, random naked stars aren't so good. They're pretty dangerous. I will just do a little bit. I'm not going to type the full things, but I'll do SRX star, and that's a little more targeted as to what we want. And now let's copy them to your space. Remember, we have this work shell variable, which has the name, the path to your local scratch space, and then there's day one subdirectory we've already made there. And so do that, and it should start copying these Fastqc reads to your personal space. And why don't you send me a chat when you got that done? Or even if you got it started. Right? So. Oh, you even beat me. I also like the ls-l. That's the long listing that gives you the last modified date, the byte size of the file, who owns it, the group that owns it, the permission bits. I generally like to see all that information all the time. Okay. All right, I got one person that says they got a copy. Anybody else got a copy successfully? Great. Okay. Great, 3 people. All right, so we're getting there. All right, so next slide. Next thing to learn about Hoffman is its package system, this module command. Okay, so let's type module, and you'll see that this module thing has all kinds of commands and stuff. A lot of commands, if you give them no parameters, give you some kind of help, more or less comprehensive, at least some kind of hint as to what you can give to it. So the main point of this module thing, now you don't, this is stuff the administrators have installed on Hoffman. You don't have really control over how recent that stuff is or what versions it is. It doesn't always have what you want, but it's got some things. The main point of this thing is to manage various Unix environment aspects like the path and other environment variables so that they can have multiple conflicting parallel versions simultaneously installed and you can choose from them. And also have some sort of dependency resolution. So if a given package depends on other packages, it can activate the environments for all of them together. Anyway, so this is we're going to use this several things. It's not always going to help us, but we need to know about it. So you can see what modules you've currently got active with module list. ATS is just the kind of the default environment for the people that run the cluster. That's them. Intel 1804 is one of the compilers, C compilers, so that's fine. That's nothing. We don't, that's always there or whatever. That's no problem. Okay, one question is, what do they got, right? So that's module avail, okay? And that'll take a second, and it will give you a fairly long list. This is stuff that's there. It kind of looks long. It's not that long because it's got a lot of different versions of the same stuff, and a lot of stuff you want there is not there. But anyway, that's the list. Not particularly easy to find whether something's there or not. So we're going to do a little trick here. So you may know the grep command where you can search for things. So I want to use Fastqc. So let's see if fast is in there. And the grep-i, the I means case insensitive because I don't know whether they capitalized it or not or whatever. So I'll just see, do they have the word fast in any capitalization in there? Well, that didn't really work because that just got me the same list as it is. And it's because the pipe does standard output to standard input, and the module avail doesn't write its output to standard output. So we got to do a little shell voodoo here to connect standard error and output to the same thing, so the pipe receives them both. And now our little grep will work. And now we can see the things that match fast. And well, they got 4 things with the word fast in it, but none of them are Fastqc. So module doesn't have it. Well, so we'll have to install it. But Fastqc is easy. It's a Java thing. It comes nice and tidy. There's already a Java installed in Hoffman. There's already a Java active in the default environment, and it's a perfectly suitable version and so on. So this is going to be an easy install. No big deal. If you open in a web browser, this place, let's go switch this for a second, you will see this. Maybe if I did the PDF right, you can click the link and it will just open in your web browser. Somebody can tell me if that actually worked or not. So this is the web page for Fastqc, and there's a download now button, link, and you go there, you should get this place. And there's a link here for the WinLinux zip file. That's the one you want, this one here. And if you look there, there's a path, right? So there's, depending on your browser, you may have to right-click it, copy link to clipboard, maybe in the status bar. Somewhere your browser will give you a way to find out what the URL of that link is. Now, we don't want to download this to our local machine. Well, you could do that. You could download it to your local machine and then upload it to Hoffman, but it's easier to just directly download it in Hoffman. So we need this. This is the path we need, and it's right here. So let's actually do the download on Hoffman in the place where, in the place where we're going to use it. So I don't know what directory we're in right now. Maybe you're still in my directory or one. So anyway, we got to go back to your place. So that's the working directory where we're doing all our stuff. Okay, so that's here. I'm now in, you should now be in your equivalent personal place and not my place. Okay. So we're back. This is now your copies of the files. You can do anything you want with those. They're independent of my copies now. And we're going to download the Fastqc zip file directly in Hoffman. So I'm going to directly copy this. Hopefully you have some. Hopefully the PDF or something lets you copy something that's reasonable. Sometimes the PDF has funny characters in it, and there are all kinds of problems with copy paste. And pasting random things into terminal windows sometimes gives really nice accidents. If you've ever copied a huge document and pasted it into a terminal window and see all those to be tried to execute as commands, and your heart stops and you don't know what those are doing. If it hasn't happened to you yet, it will happen to you in the future. Anyway, try to be sure about what is in your clipboard before you hit paste into a terminal window. So here you go. wget single quote, blah, single quote. This is all one logical line, no white space or anything. And wget is a nice tool that understands HTTP, FTP, all kinds of stuff, and it will basically download that and put it in the current directory. So this is a fairly small file. And now we have this Fastqc zip file sitting here. And we want to expand it. We're not in the GUI here, but there's the command is typically unzip. And I can just type F tab complete. That's the zip file. And there we go. It made a bunch of crap. Okay, so now we have this Fastqc directory, which contains the expanded version of the zip file. I didn't do this in the notes, but maybe I don't really like the name of this zip file because it doesn't sort with an expanded place where they put this Fastqc. Maybe I'll rename this a little bit just to have tidier space here. Now they're next to each other. I probably also personally would rename this Fastqc to have the version number on it. And maybe I'd put it in my home directory or something. But anyway, I'm not going to change it now because the rest of the slides depend on knowing this, what this is. So, but anyway, this depends on how tidy you like to be and so on. Okay. Some people might also, for posterity, you might also, depending on how you keep your lab notes or something, you may actually put a little record there of the command and what date you retrieve. You know, you might even take this whole, this would be big enough. Control D is EOF there. That will stop that. And now we have a little log file that tells us what we. There you go. That's a way to take sort of lab notes or something that you can come back to and reconstruct what you did. So now I have a little file that has my notes as to what I did and when I did it. And that's how we got that zip file. Okay, so everybody got this Fastqc directory expanded? Okay, one person. 2, 3. Okay. Let's look inside there, see what we got. We don't know what we got yet. All right, we got some subdirectories. There's an install txt. That sounds promising. Some README. Those are also generally promising. There's actually something that's called Fastqc that looks plausible for what we would run to start it. The .bat is probably for Windows or something. The jar files are Javaized zip probably contain the actual code. All right, let's look at this install txt. So less is the nice program to use to read potentially large documents on the screen, one screenful at a time. So use tab completion here to save yourself some typing. Okay, there we go. So that's the less Fastqc / install. It's probably on the next slide. Yeah, there we go. And there we go. Now we're reading the install notes. Now, first thing to know about less, if you've ever been trapped by it, is how to get out of it, and that is the Q key. That is how you get out. That's quit. All right, I'm back in. How do you move around? Space bar moves you to the next page. The B key, B for back, moves you back a page. Okay? The arrow keys move you a line at a time. And for things with wide lines, left and right, also move you left and right. Okay, that's more rare, you need that. So anyway, I'm back up at the top. And I'm scrolling down to Linux and said, blah, 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 you need a Java and so on. And like I said, Java-version, we already got a Java. Okay, there we go, blah. Okay, ready, all right. All right, sounds like kind of ready. What's this Fastqc thingy here? This looks kind of like the command name. Maybe if it's a binary, less will complain a bit and says, this doesn't look very much like text or whatever. If it's text, it'll let us see it. No, it's actually text. It's actually a Perl script and so on. So it's definitely something you're supposed to execute. That's probably the way you start this thing. So we can try it. Okay, let's try to run it. Okay, so now I'm giving the relative path here because this thing probably isn't on. It's almost surely not on your path. So we can, in the shell, you can always give an absolute or relative path to the command, even if it's not on your path, and the shell will be able to deal with it. So here we go. Oh, permission denied. Does anybody know what the problem is? I mean, from the screen that you can see right now, you can see what the problem is. Why the shell gave me this complaint? What's the problem? Why I get this permission denied when I try to start this thing? Yeah, it doesn't have the execute bit set. Unix has got these little bits that are associated with each file. Yeah, we need to chmod something here. So there's a little place right here where there's no X. So this file is not marked executable. And either the zip didn't communicate that information or it wasn't executable when they zipped it, or for whatever reason, we didn't get the X bit on this thing. So we need to get it. Now, there's a lot of ways you can do this. Some people would do this or whatever. The way I like is the relative change. For the user, the owner of the file, I want to add execute. And the file that I need to do this to is, so that adds the X bit for me. You know, there's three, RWX, RWX, RWX. The first one is for myself, the owner of the file. The second one's for the group. And the third one is for everybody else. And this file is only marked RW right now. No, the group and other have no permissions at all. It doesn't really make sense to give them an X. The only one that needs an X is me. And so I need to add myself to U plus X. And now if I look at this ls-la for this file, you can see that it has the X now. So it is now marked as executable. Yeah, so the shell will be much happier now. Unix will be happy with it now, and we can run it. Let's see if it works. Oh, what's up with this? It wants to start an X11 display. So it's got a GUI. So if you read the documentation, it turns out if you run this thing with no arguments, it tries to start a GUI. If you give it arguments, then it processes those as files. Another way is to, if sort of running the command by itself doesn't work, you can always try things like double dash help, double, dash help, dash H. Usually one of those will give you something. I think it's, let's try double dash help. Oh, there we go. Okay. So there is sort of more the usage intended for command line. This one executable is both designed as a GUI program and as a command line program. Without arguments, it runs as a GUI program. So here we go. This is the little mini help, and it's basically you just give it command line arguments, which are the sequence files you want, and then there's a whole bunch of arguments that are kind of basically all optional. There's really not even that many parameters to this thing. And so all we need to do is start feeding it some of these files, and we can actually feed it to them all at once, and it runs in parallel, I think. So let's see here. Let's scroll. All right, we just did this slide, and then we're kind of on this side. We're actually going to run this thing. Now, if these files were huge, like a full human genome, 60 times larger, or if we had a lot of them, it would not be a good idea to run them interactively in the same session that our interactive session is in. We would have to job submit them or whatever. These are small. Our interactive session is actually decently meaty. We have authorization for 4 cores and 20, whatever, 28 gigs of RAM. Let's just do it in the session. When we get to bisulfite alignment, we will practice that as a job submit and use the job submission thing. But for this early stuff and so on, we can just do it right in the session that we've got right now. And so the command would, one command, there's lots of ways to do this. So Fastqc takes a threads argument, which allows it to run in parallel. Like I said, we're off for 4. The documentation documents that each thread needs about 1/4 gig of RAM, and so 4 of them is only 1 gig of RAM. So yeah, it was way under our authorization for this session. Then you just give it the file names, the Fastqc file names, and they can be gzip. It's perfectly happy to work with gzip files. And so we'll just do everything. SRX star, that would work. This is a little safer if once you've been working with certain files of more types here, maybe not all Fastqc files start, maybe not all SRXs start with Fastqc, our Fastqc files anywhere after you've been looking here a while. This is a little safer. And just because we're interested in how long this command takes, you can always add time in front of a shell pipeline, and it will keep track of start and end and tell you at the end how many seconds it took or whatever, just for giggles. So let's do it that way. Okay, so we're going to start that off. Okay. And there it goes. It started 4 in parallel, threads 4. And it gives us actually fairly nice little progress updates about what it's doing. And it's thinking. Drink some coffee. Maybe check your phone. Maybe you're on your phone. I don't know. Analysis complete. It's probably started there. It starts the analysis of the 5th one. We got 6 to do and 4 in parallel. So night 2 got to wait for round 2. Analysis complete. Another one will launch. There we go. All 6 have launched. 2 are done. There's another one done, 3 done, I think. Five, if we're keeping track. And I actually still get messages from 2 different ones. We got 2 left, I guess. These are a little behind because we started later than the others. We've got 1 left. There we go, done. 1 minute and 50 seconds on the wall clock. User counts each core separately, so we used 5 minutes, 3 seconds of CPU time, which was distributed over 4 cores. So, and basically very little in the operating system. Okay, anybody else finished? Nope, great. Fastqc is about as easy as it gets. Basically, the only arguments it needs are the sequence file. Very little parameters. Okay, 3 declared finished. Okay, great. Let's find out what it made. Okay, so if you look, we've got some HTML files that showed up, and we got some zip files that showed up. And that's probably what we're supposed to look at. And given the HTML there and the zips and so on, probably we're supposed to look at them in a web browser. Well, so what we need to do now is we need to copy these to our local machine. So hopefully, as part of the emails I sent out on Monday, you reminded yourself or got installed or whatever, some mechanism by which to transfer files between your Hoffman spaces and your local machine. And you need to do that now. So by whatever mechanism you got, you need to move these, need to copy these HTML and zips to your local machine. Now I'm on Mac, and I will show how I do it. And you can do it all in the command line very easily. So first, I'm going to make a place to put these. All right, now I've got 2 terminals open, and I got to stop and start sharing them because I think it may not have that other. I got to add my other terminal window now to the mix here. So this one, I need all 3 of these. Okay, so there's this new terminal window, which is not in an SSH session to Hoffman. This is my local machine. Hades is my local machine. So I'm going to make a place on my GUI desktop to have, I already got, because I was playing with that this morning. Okay, just a second. I want to move the one I got there out of the way so I can be fresh and do it exactly as you're going to do. Okay, there we go. So I'm going to make a place on my GUI desktop on my local machine to put this stuff. Okay, and then I'm going to switch to it. Okay, so it's empty. It's sitting on my GUI desktop, which I'm not going to show you because it's very messy. I got icons all the way over to the left. Okay, so this is the place I'm going to put the stuff. So I need to copy them from Hoffman. Now here's how I do it. SCP command, which is kind of the CP, the SSH to CP command, SCP, and I have to tell it from what machine. You can prefix paths with blah blah blah, something at machine colon in SCP speak. That means that's the username at machine. I want you to. The next thing I mention is from there, so that'll be uscratch. I'm copying from uscratch C focus day 1. And what do I need? I need all that HTML and zip stuff, and I don't want to really type out all the paths. So I kind of want to do this. Well, I could do this. There's a couple of refinements here, some shallow magic that I'm using here, which is quite convenient. So I can't really do this because this star will be expanded by my local shell. And there are no files in my current place that start SRX, and my local shell is not going to be able to do anything there. I'll just leave it alone. I need this star to be expanded on the remote side. So I need to quote it. I could do this. That's one way to quote it. A backslash to quote a single character also works in shell speak. So this will protect this star from my local shell. It'll go off to the remote shell. The remote shell will be the expander of it. And that's what I want, because I want that only the remote shell sees in this place and knows what these file names are. So this is a way to get the remote side to do the expansion. The other thing is that I want to do both the HTMLs and zips, and I don't particularly want to type this huge long business again. That would be one way to do it. But here, shell has a magic, which is if you use curly braces, they expand to all combinations with the curly braces. Okay, so the curly braces come first, and then any other shell magic like stars and stuff operate after that point. So I mean, you can really get complicated with this stuff. You can even nest them. All kinds of good stuff can happen. All right. So let's get back to where I was. All right. And I want both HTMLs and zips. Okay. And I also want the curly braces expanded on the remote side. And the reason is because that way, if I do them on my side, I will have three, because I have to also put the local destination. So this dot means current directory on my local machine. So I'm copying from this Hoffman2 place to this current directory, current working directory, this day one subdirectory on my GUI desktop on my local machine. SCP is really expecting 2 words, from to. If I do this curly brace expansion on my machine, it'll turn into 3 words, right? This POCUS S Hoffman blah, 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 SRX, protected star HTML, and blah, 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 protected star zip. That doesn't work, then I'll have 3 words. It won't be right. I need the curly braces to expand on the remote side as well. So I need to protect those. That's 2 more backslashes there. So I will do this, and that will copy all of those to my local machine, no problem. What the remote side saw was this, essentially. Okay. So if I go to the remote side, essentially what happened is the remote side got it without the backslashes, because my shell took away the backslashes and didn't pay attention to them. The remote side saw this. And if you look, that expands into exactly the list of files that I want. So hopefully everyone's following that. I mean, there's a lot of shell stuff to learn and so on, but some of these things this is extremely helpful in becoming efficient at working. So there we go. I'm now on my local machine, and I have a copy of those HTMLs and zips. All right, next trick. I want to expand all those zips. Well, I could go to the GUI and select them all, shift click them, and open them in some zip expander or whatever. But, we're command line people now. So I'm going to do a shell loop. Okay, I'm going to do for all the zip files one at a time. I will make the path go into shell variables capital Z. I will unzip it. Okay, and then I will remove that zip file. The double dash is a little safety thing. If we have time, I'll explain it. The F means even if the file is marked read-only, get rid of it anyway. The V means just print out each file as you delete it. That's just sort of a confirmation that you've deleted the right thing. If you do an accident, at least you'll have a list of the files that went away. And then we can close the loop. So Z, shell variable Z will be bound one at a time to the expansion of this, which is the zip file names. For each one, we will first run the unzip command. Then we will get rid of the zip file since we don't need it anymore. Okay, and so bang. All right, now if I look, all the zip files expanded into directories with the contents of the zip files, and the zip files are gone. If I do a recursive listing, dash capital R does recursive listing, L will make it a long listing. You will see that, okay, that's the directory we were just looking at, and now we're looking at some of the subdirectories, right? So this is one of those zip expanded subdirectories. Well, there's an icons subdirectory, an images subdirectory, some HTML stuff, some blah, blah, blah. There's PNGs in those image directories, and each one of those kind of looks like that. We've got all this structure now. Right? So great, we're about ready to view it. So let's open it up. We could go to the GUI, double click these HTML files or whatever, but why do that? We're just going to do it all here. I can see the open command in Mac OS and also many Linux GUIs work the same way. I think even Windows has an open command these days. Will open whatever file. It's essentially like double clicking the file in the GUI. Your OS has some mechanism of guessing from the file extension or various other metadata what programs is associated with files of that type, and this will open that. For me, this will open it in the Safari web browser, but actually on this machine, I like using Firefox more often. So rather than start.html in my Safari, I'd rather it open in Firefox. And on the Mac OS, the way you tell open to override what the OS default for a file is to use -A option, which is the application to open instead. And for me, applications Firefox, that is the application I would like to use to open these files. And so if I do that, you can't see it because it's not shared, but my Firefox just opened with all of those files all at once. And now I have to add this window to my sharing. Is there a way to add a window? That would be nice. I have to ask my wife. She's the expert at Zoom. She's been doing the online teaching for over a year now. So share the screen. So now I need the presentation, 2 terminals, and Firefox. Okay, so here is Firefox. And you can see I've got 1, 2, 3, 4, 5, 6 tabs. And I'm looking at the Fastqc report now for each of them. So anyway, hopefully with all that time gone by, you've all found some way to get the business done. So does anybody else have some of these reports, at least one report open of your own? Oh, great. Okay. So let's start looking at these. I don't think the order matters too much. So this is, now, interestingly, so these ones I've chosen, they're little older runs, and Bisulfite's always been hard on the machines, as I said, and so on. So they actually, you see some more interesting stuff in these, more bad stuff than you might see from, if you've done these reports before for like RNA-seq and so on. And we're going to make some decisions based on what we see in these reports. Now, not everything Fastqc does is particularly relevant, and not all of these red X's. I mean, that's actually totally expected because we know the base composition is screwy in bisulfite libraries and so on. So anyway, so okay, let's look at the basics of this kind of Fastqc report, right? So first, how many reads we fed it, right? We fed it about 8.7 million reads. As I say, these are only been pre-filtered magically to chromosome 21. So the real genome is, much, much bigger. These files originally have about 400 million, 500 million reads. Each of them was run on earlier than NovaSeq technology. They were generally mixes of like 7 or 8. I don't know what they were. It could be GA2 or it could be, probably not even high-seq, probably GA2 millions or something. Anyway, so that's how many reads we stuffed in. And the one important graph is this one, which shows you, well, maybe first let's maybe scroll down to this, which is, I don't know why this one isn't first. Sequence length distribution. First, how long were these reads? We know there's 8 million or whatever reads, but how long, how many nucleotides in each read? 101 bases, 0 and 0 adjacent. So it looks like all these reads were, all exactly 101. So this was a single-ended 101 base run. Often, people might say 100, but maybe you ran 101. The reason is on the Illumina platform, when you have a base, there is corrections from the base before and after in terms of the modeling of the signal that you get in this base, and that's required to sort of deconvolute some messiness that the 1,000 copies of polymerized polynese, all the molecules in the amplified version of your molecule that the sequencer is sequencing. They're not always in sync, and so you sort of need to do this deconvolution forward and back. The reason why if somebody orders a 100 base run, you often run 101 bases, so at least the 100th base has a base after it to get some information back in. course, no matter what link you run, it has to be a last base, and that base only gets corrections from one side. But they asked for 100, you gave them at least 101. They can't complain. Anyway, so that's why often you'll see 101 instead of 100 or something. So anyway, all these sequences look like 101. So at least we know kind of how long the reads are now. So now we understand the x-axis on these charts. These reads have 101 bases, and Fastqc by default does this kind of odd grouping of things to sort of, why it doesn't just do 1 to 101 as far as the positions go, I don't know. There's actually a command line option in there that I mentioned that you can no grouping, double dash no grouping, and should make it just sort of do a straight through. But the default is to sort of do this sort of auto weird grouping. But anyways, such as it is, if you don't like it, you can just do this command line option, double dash no group to the Fastqc run, and it should change. But this is basically a box plot that shows for each position in the read the per base Q scores. Each base from the base caller has a quality score that's Fred scale from the original chromatograms when people used to do Sanger sequencing, or people still do Sanger sequencing. So the scale is logarithmic. It's basically 10 times the log base 10 of the probability of error. So Fred 10 is a 90% confidence base. Fred 20 is a 99% confidence base. Fred 30 is a 99.9, one chance in 1,000 being wrong. So on the Illumina platform, Q30 plus is common and very good. Q20 is not that bad. I mean, it's only a 1% chance of error. Below Q20, not so good. The modern platform doesn't even distinguish many quality score values. It's almost essentially there are things near 0 and things near 30, and there's only like 3 or 4 scores they use anymore. But these older ones are more continuous for the quality score estimation. Anyway, this one looks okay right now as far as this goes. These are all kind of in the 30 plus range. Maybe the beginning, it's not that uncommon. The first few bases are a little wacky, but okay, that's not really disturbing. Physically, on the flow cell chip, different spots of it can have various physical problems and so on, bubbles in the flow lines during the run, whatever. Anyway, this shows you sort of the distribution of tiled trouble by cycle, and basically this is a no trouble cycle, no trouble anywhere at any cycle. This is the average Q score per, for each read, find the average Q score, histogram that we know that's all high. So far, no problem. This is a very, very important chart for BS libraries. So this is for each position in the read, the ACGT base distribution. So line up, stack up all the reads, go to position 37 or whatever you're interested in at the moment, and just take all that pile of ACGTs from all the reads and histogram it, right? So what we see is exactly what we expect from Bisulfite library in human. The C percent in blue, very, very low, very, very depleted in Cs. They all got turned into Ts. The A's, which are in green, are here. Normally A and T would be roughly the same, right? Because C's, you're sequencing random strands, right? C's and G's pair and A and T's pair. So CG, C and G should be all the same, and T should be all the same. But no, the T, which is in red, is above the A, which is in green. And that's because the blue went down and the T went up due to Bisulfite conversion. And then the G's are, wherever they are. So this looks good. This is exactly what we like to see, and there's not a lot of dependence per cycle. Pretty flat. That's good. It's a little oscillatory, and that's not ideal. It means some part of like the prephase and phasing calculation that the base color isn't quite right, but this is tolerable and it's not that unusual. So this one's looking okay this way. This is some stuff about GC content. This is if there's any uncalled bases, ends in there. Basically, these are end free pretty much everywhere. We already looked at this one. This dupe plot is important. This is. I mean, there's many ways to estimate this. Fastqc has a particular way, but basically, when these things are high, it suggests that the library is of a low complexity with many PCR dupes or whatever. Maybe there's some here, but so it's beyond the scope of the workshop to deal with dupes. And this is another very important one to look at, adapters. This is, are the inserts so short that we run into adapters? Fastqc does this by having a database of commonly used adapters. It doesn't know them all, but it's got most of the common ones. And it just looks for those k-mers of these known adapters, and is telling you where those k-mers are found as a position. Are they found frequently at various positions of the read? And basically here, none of these common guys are showing up at any appreciable fraction at any cycle. So this library does not look like it has inserts short enough to read into adapters. It looks fine. So this one looks pretty good. So this is the 116604F2. So that one looks fine. Okay, so let's look at one that's less good. Okay, I don't know. Let me just scroll around and find something. So here's 101207, SRX 101207. Those are the NCBI SRX experiment numbers, which is where I got the data from, the NCBI short read archive. So this one, okay, we've got 6 million reads. What kind of lengths are we dealing with in this library? Well, somehow there's been some kind of trimming or something that somebody's already done. Most of them are at 100. There's some 100s, there's some 102. Well, that 102 looks like a 0 and the 99 looks like 0, but there are some 100s. So anyway, some are 100, some are 101. Maybe somebody tried to trim adapters and there's some little false positives and ones. Anyway, whatever. It's partly 100s, partly 101s. It could also be that it was a mix of runs. Most of these, as I said, are a mix of like 7 or 8 runs. Maybe some of the runs were done as single 100, some of the runs were done as single 101. That's actually probably the most likely explanation. Anyway, but basically these are 100 runs. Okay, what do we got? Well, here clearly by the end of this run, the Q scores were taking some damage. All right, so maybe the tails of these reads aren't so good. And some of the tiles weren't too happy towards the tail end, and that's probably, these bad tiles are probably dragging down this box plot towards the end. And now look at the per cycle ACGT distribution. Well, we've got the pattern where the C is low, the T is higher than the A, but we see some cycle dependence, and that's not good. So here we see there's some sort of anomaly kind of at the front, and we see also that the %C is kicking up at the end. This makes me want to pass to a subset of cycles, right? So maybe, we start somewhere on cycle 10, 11, that would get rid of this crap at the front. And we got to wonder what's going on here around cycle 80 and after. Most likely explanation is we're starting to read into adapters which have a brighter C distribution than the BS library does. So we can check the adapter figure down below to see if that guess is right. Here's the end content, nothing much to worry about there. We already saw that one. Dupe levels actually look better than the other one. Adapter content. Oh, it's Illumina universal adapter. Starts to kick up. Those k-mers are starting to be seen around cycle 70 plus and so on. So yes, probably that's the reason why the C % is starting to go up, because we're actually, if some of the fragments are too short, we're reading off into adapter. The adapter has C's at a more normal level than BS library, conversion library does. And so that's it. So if we wanted to deal with variable length reads, we could run a program like cutadapt or something to actually recognize that Illumina universal adapter and variably cut where that starts to the end. A simpler approach, which is what I'll take in the workshop just for simplicity, is we'll just cut. We're just going to kill the bases from 70 something, on or whatever. They're not very high here, and towards the end, it's 5%. It's not crazy, but anyway. But we'll just take a subset of cycles to deal with the problem. Anyway. So anyway, that's the kind of things to look for. That's the kind of things that can happen. I think the kind of looking at those two, you've probably seen the kind of stuff that can happen. Here's 043407. It looks like the tail end of this guy is not too good. So some of the tiles just kind of, took a, they just, who knows? Some of the tiles died. Again, we see the front has a little bit of an anomaly, not as bad as the other one. Oh, we're kicking up the %C there at the end. Per base end content goes way up. So actually those tails of the reads probably aren't dangerous in the sense that they contain ACGTs that are wrong. Looks like the base caller just ended them out. It wasn't too confident. So anyway, but they're, but it is 80% of reads or something. So almost, 4 out of 5 reads have gone to ends by cycle 74 or something. So that tail isn't too useful. This is actually a mix of a little bit of 85 MERS and some 100 MERS. Dupes don't look too bad. Even the adapters don't look too bad. Anyway, but maybe we'll do some trimming here. We were planning to trim the other one down a little bit anyway, because he's got the adapters at the end. Maybe, if we trim at 74 or something, that this guy's not good past there anyway. How about this guy? Yeah, the tail end maybe a little bit. Okay, a little bit at the front. Same thing about cycle 11. Starts to normalize a little bit anomaly at the end. How about adapters? Yeah, adapters starting to kick up around the 74. Okay, how about this one? Find the read a little worse than the error, but that's still 30 plus, no big deal. 1 in 1,000 error, no problem. Okay, the very end, not so great. Some tiles, exploded.
Hi, Sean.
Yes.
Yeah, can I ask a question, a practical one? So, say you do a big sequencing experiment like this, and you've got, I don't know, 20 samples, and then when you're looking through the QC reports, there is that variability. Is the best practice, therefore, in order to try to get as much meaningful data out, to trim away as much of the ends as possible, so that now the quality of the reads are useful or have high enough threshold score?
So yeah, so I mean, there's two things fighting, right? Which is to have uniform, high quality, and ideally same length for everybody. Because remember, the longer they are, the more of the genome is bisulfically unique, alignable, and less of it isn't. So if you want all libraries to sort of have access to the same sites, then the lengths have to be comparable. So ideally, we kind of would pass to a high quality subset of a high quality common length subset for everybody. But the thing that's working in the other direction is that every cut we do is going to kind of, your coverage goes down. And if we start cutting out 20% of reads, well, there goes 20% of our coverage, and coverage is kind of precious anyway, right? I mean, we're not really having an overabundance of coverage. Maybe our, I think in these libraries, it's like the means are like 4, 5, 6, 7x for human. I don't know. It's not generous. We're not. It's not 10. It's not 20,
Sure.
and we're throwing data away. So we're going down. So those are the things that fight, right? So.
But I'm just wondering if you had, say, an experiment with lots of different data sets, and then say 20% of them require you to trim away a substantial amount so that the read lengths are, say, 80 versus the remaining 80%, which are good across approximately 100 bases. Is it fair, therefore, to combine the analysis looking at samples that have different read lengths like that?
So 80 versus 100, not too bad, right? Because the uniqueness is more. Uniqueness usually goes through a phase change, right? So if you look at k-mers of size, 1, obviously that's a single ACGT. That's as not unique as it gets, right? And then you go up to 10 mers. Well, 10 mers are everywhere, right? I mean, there's only so many different 10 mers, not that many, 4 to 10. But by the time you get up to the 20, 30, now there's supposed to be a phase change, and you go from basically no k-mer unique to almost all of them unique, right? And the transition zone usually isn't very wide. Now for regular like RNA-seq and stuff, the transition maybe in the 30s. It depends on the genome. Obviously, Arabidopsis will transition before human. But 50 people don't run shorter than 50 these days. So you're way in the RNA-seq uniqueness range there. You're also kind of already in the bisulfite uniqueness transition zone by there. So by 80, you're kind of well on the right side of the phase change usually. And so 80 versus 100, not too bad. So if the quality is really junky in that bases 80 to 100, yeah, maybe it's a win, right? To get rid of it. It's all, and it depends. In mammals, you're more concerned about CG methylation, which is more an on or off thing, and not too bad. If you're in plants and dealing with CHH methylation, that's your concern. Well, that's maybe only 5% when it's high, and now the particular little contaminants and so on in the methylation files start to be a big fraction of the methylation signal there. So yeah, mammals 80 versus 100 CPGs probably okay.
Okay, thanks.
But yeah, I mean, a lot of things come down to the particular regime that you're in, and different genomes don't have the same statistics. Yeah, so anyway, so what I'm seeing from looking at these reports is that, and given that we kind of want to keep things simple here, what I'm thinking is, in this case, to maybe cut off the first roughly 10 bases and cut off somewhere from the mid 70s to the end, and maybe get them all to be some common length, maybe around 65 or something. Right, 75 minus 10 would be 65. So anyway, so that's the plan. Okay, so okay, we'll go back to that one in a second. But here's the plan, right? So we'll keep it simple here. For all 6 samples, we're going to keep base 11 to 74, which is 64 NT for all 6. Okay, just it's easy to do. I mean, we need to move on, right? I mean, it's already 11:15 on day 1. Right? We want to get a little further. So let's just make that as our decision. But you kind of see the things that we think about in the question that was raised and so on. This is the kind of stuff that goes on at this stage in the thinking that happens. Okay, just for some more background on what these reads particularly are, it's this Nature paper from 2013, charting a dynamic DNA methylation landscape of the human genome. It's a Meissner paper. These are human whole genome bisulfite BS reads. I got them from NCBIS Array. This is from an era when BS-Seq was younger as a technology, and the Luna platform was less mature. The originals had 350 to 500, 600 million, 350 to 600 million reads. They were variously single-ended, 85 to 100 bases. The one was a paired end run. There are roughly 5 or 7 of these older selective platform lanes merged together. The first one is two bioreps of H1 embryonic stem cells differentiated to neuroprogenitors. The next pair is 2 bioreps that are differentiated instead to mesenoderm. And the last one was actually a paired end run, only 1 biorep. These are the ones differentiated to mesenchymal. I needed sort of 2 bioreps to kind of make a nice design for the rest of the analysis. It was, I wanted some symmetry, 3 tissues, 2 bioreps each. So I faked this one. I took the PE apart. I made main end 1 be fake bioreplicate number 1, and I took the second main end, had to reverse complement it because we want it to look like CPOR, like the rest of it, and called that bioreplicate number 2. Obviously, you would never do that with real data, but it makes for a nice test. I'm trying to put together some biologically interesting but small test case for us to work with here in the workshop. So that's what I did. The way I cut them down to chromosome 21 is I did the full analysis with the full genome BS bolt alignment to the full human reference. That gives me a BAM alignment file where I can see what reads align to chromosome 21. I took the names of those reads, went back to the original files and pulled out those reads and made subset files that are just those. So we know sort of a priori that they're now all going to align just to chromosome 21. And that gets the files to be a much more manageable size because we can't wait for hours while alignments. We're going to need to do operations several stages. And we need files that are small enough that we can get the operations to complete relatively rapidly. So that was how the sample data for the workshop was constructed. Okay, so this trim to 11 to 74 decision will more or less take care of all the problems we saw in the Fastqc report. Takes care of the adapters at the ends, the per cycle ACGT instability, the lower Q-score of the tails, the tiles that are dying. I forget if one of the libraries I think may have even had, it looked like something wacky happened, like some of the 8mer index reads that are part of DMUX got actually attached to the end of main end 1 or whatever. Anyway, those will be gone. We're going to stop at 74. Those are out there at 102 to 109 or something anyway. And we're also going to, this also had the side effect making all the samples have the same uniform length. And so all of the samples will have the same BS uniquely alignment accessibility to all, which is nice if it's not trading off, things that are worse. This costs us some coverage, actually a significant amount of coverage, right? I mean, some of these were 100 MERS and we're now down to 64. We've thrown away a third of the coverage for some of these, worse for the shorter one. So this is a pretty steep coverage hit, and maybe if I think the original analysis didn't cut it this carefully, right? We would probably try to keep more. Maybe those first 10 cycles really aren't recoverable because you really don't like that instability of the percent content. That doesn't really make sense. But the tails we might try to rescue more. There's no sense in condemning all the reads just because some fraction of them, even if it's a significant fraction of them, have problems with the tails. We would probably try to rescue the ones that don't, and try to get the coverage a little higher because the coverage is too precious in this kind of. We don't have an abundance of coverage in this study. By passing to I've given you a subset of all the reads that align to chromosome 21 and not a random subset of reads of the same size. That is because I want your example to have representative coverage of the whole data set, right? If I had just randomly selected a subset of reads of the same size as I gave you, they would spread to all over the genome and the coverage everywhere would go down. And then we can't really do analysis because the coverage per site would be so low. So I needed to, essentially I've done an in silico targeted bisulfite that just kind of pulled out chromosome 21. But at the same coverage as the original study was. So this is the coverage that you will see in this example is representative of the original study, apart from the fact that we killed the coverage a little more than they did. And we're not going to deal with the dupe issue in the workshop. We just don't have time. Okay, so how are we going to affect this cut to 11 cycles 11 to 74? Well, it's not too bad. I mean, it's simple enough we could really hack it up with some commit shell, wizardry and some Unix command line tools like awk and paste and cut and all this stuff. But let's use a real tool that's designed for it, because this is also the tool you're going to use to do adapter trimming, which isn't something you're really going to hack up in a quick shell, shell pipeline. So cut adapt is one tool to do this. It's very commonly used. And we got to see if it's on Hoffman, and if it's not, we'll install it. So okay, this is my window for my local machine. I'm now back to my window for Hoffman, and it's been so long. Let's figure out where I am, and that is my personal scratch space copy of the working files for the day one, right? So this is where we've been working. This is where your Fastqc report should be. All right, let's see if cut adapt is pre-installed on Hoffman. Yeah. Let's do that. Okay, so we'll do our module. We just space. So let's do our module eval trick to see if anything has the word adapt in it. And there's adapter removal, but that isn't cut adapt. So the answer is no, it doesn't look like it's pre-installed. It's a little more tricky than that, because cut adapt is actually a Python program, and many Python packages are managed in a internal system, a subsystem, sort of a whole sub-universe, another package, package managers. So it's package managers all the way down, basically. So we really need to ask the Python package manager whether it knows about this thing. So we need to know what kind of the Python situation on Hoffman is. So let's ask, okay, what about Python in the module system? Well, what flavor do you want? Do you want a 2 flavor? Do you want a 3 flavor? Those are incompatible, unfortunately. There's several versions of the 2 flavor and the 3 flavor. There's the Anaconda version. There's all these. Python, more than most systems, seems to have many environments, installers, packagings, alternative package managers of it, and so on. And so there are many, many, many environments that you can get access to Python in. And many packages are available in many of those choices. And so there's not usually just one way to kind of do the mechanics of getting a package going in Python. It depends on what kind of your local environment situation is. We will just kind of do base flavor Python on Hoffman. We will do the Python 3 series. That's the more recent series of Python, and cut adapt is good for Python 3. And so we will, there actually is a 3.7.2 on Hoffman, but I've done all my testing with 3.7.0. So why don't we all load 3.7.0. So this activates this particular, you see we have many parallel installs of Python on Hoffman. This here we're designating, I want the base 3.7.0 flavor. And this has done all kinds of things into my shell environment. It's the shell environment so messy it's hard to see, but if I show my path or something, you should see some Python bits in there. Yeah, see, so now there's some Python directories in my path and stuff. Anyway, so that's activated stuff. So now we can actually ask the Python, one of the Python 3 package managers, pip is the package manager that's one of the ones convenient to use on Hoffman. And now that we've got Python activated, we can use pip. Pip3 is a little less ambiguous. It unambiguously means the pip for Python 3. And pip3 list will list the packages that the Python manager, package manager is aware of being installed. And for me, it's actually a list of some 100 or whatever. We got this same issue. It's kind of line of scan through. Let me see if adapt is in this list. Oh, it is, because I've previously installed it. But I'm sure when you run this, you do not have cut adapt installed. Maybe you do. I don't know. Then you may not need this workshop. But if you don't have an install, it's pretty easy to install because pip will do all the work for you. Okay, so if you don't have it yet, you say pip3 install cut adapt. The problem is that that'll try to put it in the system directories where you don't have permissions. You're not an administrator on Hoffman. You have to give this --user option, which means put it in one of the places I control. So here's how you can install. Many of the issues on shared Unix systems are how you deal with installing packages when you're not an administrator. So it's relatively easy. With some package managers, it's relatively easy. With pip3, it's just a --user, and then it redirects everything into subdirectories of your own. In particular, it's in your home directory. the .local subdirectory is where stuff's sub stuff. We'll look at that in a second. But I'm not sure what will happen since I've already got it installed. Maybe it will reinstall. Requirement already satisfied. Yeah, okay. And my pip is out of date. So what? Okay. So yeah, it's just telling me I didn't have anything to do because you've already got it. But if you don't have it yet, that should have installed it. It figured out the dependencies. It looks like there's 2 dependencies, xopen and DNA.io. And it went and it downloaded cutadapt, xopen, DNA.io, and installed them all 3 together. So the dependencies of cutadapt were satisfied. And now you've got some new Python packages ready to go. And as I said, the place it's putting these is in your home directory. So here is my home directory. And there's this .local, here. There's this .local subdirectory of my home directory, and you will have one too now. It's hidden. Normally, .files don't show in your ls. That's why you have to do this -a. The -l is giving me the long listing as usual. The a, little a, is showing me .files. Normally, files that begin with a dot are hidden in Unix. If you ls-la your home directory without the grep, you will probably be surprised at all the .files that are there that you didn't know about if you haven't looked for them before. Yeah, it's the pip has put stuff in there. So if I look in there, I've got the bin, the lib, the share. If I look in the lib, I've got a Python 3.7 subdirectory, site packages. Oh, there's all my stuff. I got cutadapt, BS-Bolt we'll use later, NumPy, the other dependencies. Anyway, that's where it's shoving all these packages, and it's taking care of it for you. And you can thus install Python packages without being an administrator. Okay, and that bin subdirectory is where it puts sort of the ones intended to be executed directly as if they were commands. So in my .local bin, you see that there is a cutadapt. That is the command we're intended to run. Now, .local bin, it probably isn't in your path. It wasn't in mine. I don't think Hoffman adds that there because it's not even a standard thing. It just depends on whether you like Python or not. And maybe that's not even the place you like to put your Python package. So anyway, so if we want to run this thing, just like we had to kind of give the path for Fastqc, we kind of got to give the shell the path to where this thing is. So that is where it is installed for me and for you too, and that should run it, and it does. It runs, and it says, "Yeah, I'm cutadapt 3.2. I'm running on Python 3.7.0, and you didn't give me any command line parameters. Please give me something to do. And if you want help, --help." So there you go, --help. Now you see all these juicy command line parameters, which you can do adapter trimming with and all kinds of stuff. And if you look in there eventually, somewhere there's some stuff to trim out fixed numbers of nucleotides from the ends, and we'll do that in a second. But basically, that shows we shows this thing running. We did the install part. That's always the first hurdle with any new piece of software is getting the install done. So we did that. Okay, so to make this running it more convenient and so on, let's add it to our path. So the path is the shell environment variable, which has a list of all the directories that the shell looks in to find commands. Okay, now that is hard to read because it's a single string with colons separating the directories to look. Hard to read that way. So I'm going to use the tr command, which can replace one character with another. So those colons I'd like to turn into new lines so that each one is on its own, each directory is on its own line. How do you type a new line? Well, it turns out tr understands escaping, and tr\n, tr knows that means new line. The problem is, how do we get a backslash to tr? Now, I could put it in single quotes, and the backslash will get unmolested to tr. Or if I don't use double quotes, I got to put double \n because my shell, which sees the command before tr gets it, uses backslash as a special character. And so I need to, if I want a literal backslash to go in the argument to tr, I need to put a double backslash. So then my shell unwraps the double backslash to single backslash, and then tr knows \n means new line. Okay, so that's how that worked. And now I get a list of my path directories in an order that doesn't bleed the eyes. And anyways, there you see some Python stuff there. Oh, here's the real core system stuff, /bin/user bin, user local bin, user sbin. Here's my personal bin directory and my home directory, but I don't see anywhere, there's none of this .local bin stuff, so it's not in my path. So we need to add it to the path if we're going to use this command as if it was like any other command. And the way you do that is like this. And some people would probably try to use shorter versions of this, but there's reasons for everything I do. This is a little safer if your path contains weird characters in it or spaces or whatnot. Here we're adding the new directory at the end. That's generally the safer choice because in conflicts, if a thing is found in multiple directories, it's the directory found first in the list that wins. And system programmers and other programs are always looking for programs that, they generally start many programs themselves and they use the path as well. And so generally safest to put your new stuff at the right end so that the people looking for stuff already will never be to find the thing that you added. It's generally only a problem if you added a directory that contains like other copies of base utilities. Like if you had your own ls or something and it wasn't ls, then you really be starting to cause chaos if you put that directory near the front of your search path. Anyway, yeah, so with that added to my path, and the export means that not only is my shell going to get this path, but every program this shell invokes. So this is now inherited by all programs that this shell starts, which may or not be what you want, but generally you want to think less about path and you export it so that it's sort of more global in your environment rather than just in the shell per se. With that done, I can now run cutadapt without saying where it is, just like if it was a regular command, which is how any other regular command is ever found because it's on your path. There's nothing special about doing this. Everything that you ever type as a command name that doesn't have a path associated with it has to be done by finding it. All we've done is added a new directory to the path. So effectively, the things there get added to our list of things that we can type as commands without specifying paths. So there we go. Okay, so that's a little bit about paths. And before we run this thing, our little working directory is getting kind of messy. So I'm going to show you how I sort of organize things to both kind of keep an effective in silico lab notebook and just kind of manage stuff as you do analyses with many steps. So let's start organizing. So let's make a directory for the Fastqs that we started with, and I'll call that stage 1. That's our base. And let's move all of our Fastqs into that place. The MV-V means it'll get the line of progress as it does each one, and the I means if we do an accident and try to move something on top of something with the same name, we'll get a chance to say, you want to overwrite, yes or no. So there we go. And if I look, I now have I have this new directory, and all my Fastqs disappeared from here, and they are now sitting inside that subdirectory. So that's kind of good. That's kind of our starting data. Now we also have this Fastqc analysis. So let's give a directory for that. All right. So stage 2 was Fastqc before cutadapt. So there's a directory to hold those outputs. We haven't populated it yet. Let's populate it. So let's copy all of the HTMLs and zips that start with SRX and put them in there. Okay, so there they go. All right, looking better already. All right, so they're now in stage 2. We got those guys. And we didn't make a script to actually do that Fastqc analysis. So if we kind of, this session dies, we'll kind of lose our record of what commands we ran. So while we kind of got them in our mind, or maybe in our history, or maybe we can scroll back in our buffer here, let's make just a little text file that contains, oh boy, I might have to scroll back a ways to find that. Yeah, it was around here, right? So there we go. That was the command we ran. So this is what we ran. Now that's not quite right because we moved files around after that, so that command isn't quite right. So maybe we'll lie a little bit and edit our log of what we did to make it a little more consistent with the way files are now. I mean, it would have been better if we would have made an executable, if we had sort of rearranged first and then ran it, and then we could log our actual command. Right? But if we want to kind of do this now, this is effectively a command that should reproduce it. And if we were really serious about this, we would run this to verify we would reproduce now. All right, so I've just changed the path. We used to have all the files in one place and didn't have subdirectories. Now we got subdirectories. So now I got to say that the inputs for Fastqc are from that stage 1 subdirectory. And the way Fastqc works is we would write the results in that stage 1 subdirectory. And the way I usually let it happen is as you reach each stage, in stage n, you have the files that were produced as a result of stage n minus 1 at the beginning of the analysis, the next step of the analysis. I let stage n plus 1 produce its outputs in the stage n directory. And then once everything's hunky-dory and there's no errors and you think it's kind of roughly stable, I create the stage n plus 1 directory and then move the things for n plus 1 from n into n plus 1. And that's kind of how that's kind of the inchworm steps that kind of you ooze along your way along the pipelines. So there we go. So now we at least have some sort of little record as to what we did. Okay. All right, so now we're kind of cleaned up. All right, now we're kind of ready to do that kind of inchworm-like process of sort of working with Cutadapt. Okay, so all right, so let's run Cutadapt. Now I've already given you a nice script to do this, and this script illustrates a bunch of more principles, which are nice. And the advantage, what we're going to do this time is instead of sort of interactively running commands on the command line and then dealing with trying to record them later for there's always a problem in bioinformatics is reconstructing your steps at the end, right? You start a project or whatever, and 12 months later, when you're writing the paper and you've done 18 versions of the analysis, and you're trying to figure out what the heck the thing you actually put in the paper is, you need to retrace your steps. You got to leave little breadcrumbs all around for you to figure out how it went, right? So that's kind of what the discipline is for working. I've given you the one nice way to do it is to actually do the first time you do the analysis, actually write the script then, and you do that first time by actually running that script. And then you know that that script is the thing that produced the analysis because that's the one that made the original analysis instead of trying to take the notes later and maybe accuracy issues, especially when things are around. So you're going to grab a copy of the script and bring it to your current place. The script is currently in my Scratch directory, and you're going to copy it to where you are now. So you're currently in the working space of your Scratch pile, and now we want to copy this one shell script into your place from my place. So here we'll put the flags on at the beginning, right? So in a second. But the place you're going to copy it from is my working directory, and you can use some tab completion to help you out here. So you just got to kind of, all right. So you hear that? I don't know if you can hear the beep. My screen flashed, and you hear a beep. Because tab didn't finish because tab completion said there's more than one way to finish this. Well, if you keep banging tab enough times, it'll just list the ways that complete. Obviously, we have the BS read subdirectory and the script subdirectory. Okay, so you just have to type like S to get past the ambiguity and then tab complete. And I think there's only one file in here, so actually tab complete right on that slash should actually give you the one file name that's in there. And we want to move that to the current directory. And now you can use the arrow keys to move around and edit. You may also know the shortcut keys Control-A to go to the beginning or Control-E to go to the end. And let's go back here and put in the flags. VI is no surprise. I've used those a few times. That's the verbose show, gives you the little from arrow to as a little progress bar. The I is that safety measure. What's the P? The P preserves permissions. So this is a shell script. It's got X bits from a chmod on it. You might as well copy the file with the X bits in place. The P preserves permissions. That's what that little P in front is for. All right, there we go. All right, now you should have a little stage 2 to 3 cutadapt.bash shell script, which is the script that's going to eat the stage 2 data and produce the stage 3 data. It'll produce the stage 3 data in the stage 2 subdirectory, which once we're happy with it, we'll move into a stage 3 directory. That's how we complete this sort of discipline of working. So let's look at this shell script. There we go. And what are all these components of it? Okay, well, so I've highlighted some of them here, and sort of there's a general pattern here, which the actual meat is very, very small. It's just this. But sort of there's a whole bunch of principles that work here, which are kind of good for generalization. So this shebang line, number 9 bin bash, declares this as a interpreted file, which is to be run by the bash shell. So this is essentially how you write a bash script. You start with this. Then there's this issue of, I think some of the text in the slides talks about it. I'm not sure how much of it I got out verbally, but You know, we had to do that module load stuff and that export path stuff and whatever. All that environment-like stuff only lives in the session that you did that. Scripts which create their own shells and so on start with a different environment, clean environment. Well, some stuff they inherit, some stuff they don't. Jobs that you start with the job manager, clean environment. You know, so you need to sort of, for anything in your commands that you have in the script that depend upon maybe some stuff you did in your interactive environment, that interactive environment isn't attached to the node you're on or Hoffman or your account or anything. That's a very ephemeral thing. So in your script, you need to build up that same environment as you did in your interactive world or whatever else. So for us, that's the module load Python 370 and the export path user, home directory local bin stuff. As it turns out, the module thing is not really part of base Linux or anything. That's really something that Hoffman added. And the way they sneak it into your shell environment is to run a global script. When the shell starts up, when every shell starts up, when every login shell starts up, the shell reads certain directories at certain well-known names all over the system. Some in your home directory, some in the system directories. One of the system directories is /etc/bashrc. That is a file that every login shell reads, and it is a file controlled by the Hoffman administrators, and it sets up some stuff, including makes the module thingy accessible to that login shell. Well, other shells that aren't login shells, like these script shells, don't get that automatically. So this /etc/bashrc is a little bit of magic to say we kind of half want to be like a login shell, and we sort of need access to that stuff. We need the module thing and so on. So that sort of makes that active. Then we can do our module load stuff, and then we can do our export path stuff. And those three lines basically build up the same environment we had going in our little interactive world here. So now our shell script is living in an appropriate environment to run Cutadapt. Always good in your scripts to have some kind of progress information, like when, probably would be a good idea to say, what does this starting Cutadapt run or something. You want to have some declaration that you started, maybe the timestamp of when you started. You definitely want to have a timestamp and a declaration that you reached the end of the script, because if the script aborted early or something, especially with job submitted scripts, it can be very hard to tell sometimes whether a job submitted script actually finished to the end or not. So many errors end up being sort of silent in various ways. Having a log file that at the end of the log file says done is a great way to know that the script got to that point in the command line. If you have the start and ending timestamps, you can also retrospectively go back and look and see how much time that takes, which helps with planning future things. And then maybe some progress, progress item per file or something, I don't know, some kind of unit of measurement sense also makes sense. So these little blue echo lines are sort of some progress and sort of just some sanity checking, kind of very useful things to have generally. The main meat here is going to be a loop that's going to go through all of our 6 Fastqc input files here. Cutadapt, the main goal is we're going to take our base Fastqc files, cut out the bases we're interested in, write new Fastqc files. So it's stage 1 is the inputs to this stage really, and we're gonna produce new Fastqc files as output. So the main shell loop here is to loop over all of the individual Fastqc files one at a time in stage 1. Then this is the real meat. This is what we call Cutadapt. Our environment, we're going to run this script in our interactive environment, which has got 4 cores authorized. So we're going to have Cutadapt run as a multi-threaded 4 cores. These are the parameters that actually affect the 11 to 74 NT, whatever the, 11th to 74th NT keeping. You have to read the Cutadapt documentation to do that. You can basically get that by just saying the Cutadapt-help. That basically gives you enough information. There's also a web-based documentation, I think, for this thing. You got to figure out what parameters need, but it turns out that for what we need here, it's _cut 10. That means basically throw away the first 10 bases. _length 64, that means keep only 64 bases. And this means reads that end up less than 64 bases, throw away. So basically I don't think that ever activates in any of our cases, but anyway. I'm actually writing this long command line on 2 lines. So the backslash at the end of this line means this line continues to the next line. You could also just type this all in one really long line. Eventually your pipelines, this one gets so long, you don't really want to pack them online. Eventually you have to sort of use white space and indenting and so on to have some sanity in your complicated lines, and the backslash line continuation lets you kind of build up complicated lines where you have some control over the formatting. This is the shell variable. I put the name of the stage 1 input Fastqc in. I usually try to name the variables a little better than Z or something. This is in, meaning an input file, FQ, Fastqc, and it's a gzip Fastqc. And then this is the -O is where you tell cutadapt to write the output. And for the output, I'd like to use the same Fastqc file name, but add a NT11 to 74 suffix. But I don't want the NT11 to 74 hanging out past the fastq.gz. The .fastq.gz is kind of like the file type. I want my NT11 to 74 to show up before the fastq.gz and still have a fastq.gz on the end. So this is a little bit, one of the things the shell can do is actually do a little bit of text substitution. So you don't even need like set and off for that kind of stuff. You can actually do some of this text substitution directly in the shell. You do it the same way you refer to a variable. If I didn't have the / business, this would look just like using this value of the shell variable. But when you have the slash that introduces text substitution, this is the text you want to search for. And after the slash is the replacement. Now the replacement here is the empty string. So what I'm basically doing is taking that .fastq.gz suffix and throwing it away. So now I've gotten rid of the .fastq.gz. Now I can continue to add my -NT11 to 74, then put my .fastq.gz on the end, and now I've transformed my file names in the way I want. And there we go. So that is this script. And now you can run it. Okay, so it's been a while since we've all been together and you're running stuff now, but all you got to do is give the path to this thing. It's in your current directory, so // means here's the path to the script I want to run. It's in the current directory, and this is the name, and here we go. So now this is running in my current login session. Cutadapt is actually pretty snazzy with its little ASCII art and stuff, giving me a little progress bar. And it's almost gone through, half, well, there it goes. All right, that didn't take long. We got 3 gigs of files to process here. It doesn't look like it's going to take very long. But you can see those little triple @ signs help identify the little echo lines that we made versus all this other junk that Cutadapt is spewing out. We could have actually run the script recording its output to files if we wanted to capture this, or we can just copy and paste it in later. That's a refinement. I won't do here. We've already got enough fancy things going on here to learn about. But, all right, so how many is that? 1. That's number 1 done. That's number 2 done. That's number 3 done. This is number 4. All right, how about some chat? Are you guys keeping up? You got it working? Is it grinding away? All right, great. So you kind of following the shell script stuff. I mean, it's not all required, but you're trying to build not just scripts that do the work, but you're trying to build a whole system of working that kind of makes sense. Yeah. That'll be the last one. One more. No, one more to go. Okay, done. It didn't take too long. That took only 47:08 was the start, and 49:35 was the end, like 2 minutes. So let's see what that did. As I said, that's going to put the outputs in the stage 2 directory. So if we temporarily switch to stage 2, stage 1, right? We actually put them in the stage 1. The inputs for stage 1, we wrote the outputs to stage 1. There they are. All right, so now we're in the stage 1 directory. We've got these new NT11 to 74 guys, and that's looking pretty good. I didn't see any error messages in the in the text here. The files, none of them have 0 size. Another reason to use Allele all the time is that things like empty files are going to be very obvious to have 0 as the size of the thing. These look reasonable, we could actually start to peek into these to see if this thing actually did what it was supposed to do. All right, so let's do that. All right, so let's look inside these Fastqc files. So here's one of the input files, right? So here is an input file. So gzip, if you want to just peek into the file, we don't want to unzip it completely and take up more disk space and so on, and then have to re-gzip it later. We try to work with all files zipped all the time to both conserve disk space, network traffic, and so on. It's faster even that way to do it. You just have to be a little more sophisticated so that you can use them in their compressed form. So gzip-c-d, the -c means to do to standard input or standard output instead of to disk. The -d means decompress. So then I can give it this. So gzip is going to basically open this gzip file and start spewing out to standard output all the contents of it. But it does that on the fly without touching disk, and it'll leave the original file alone. So if I type this to less or something, you can now see the file. We can page up and down or whatever. And we can actually, scroll on this thing, and when I hit Q, nothing's happened. The file's still gzipped or whatever. We just did that all on the fly. Okay, very, very handy to work in pipelines with gzip files without ungzipping them on disk. So let me just grab. So I'm assuming you've all seen Fastqc files before. So basically, there's an @ sign, some read name, possibly with extra metadata. Then there's the actual ACGT sequence, and we've got some ends in this one. This is the actual read, 5' to 3'. Then there's on a separate line a plus sign, and then there's the per base quality scores, which are really integers from 1 to like 30 or 40 like we saw, but they've been encoded in single ASCII characters. If you say man ASCII, you will get a chart that lists all the ASCII characters. There's the decimal column. The second column is the decimal column. So you see here that like, ASCII 33 is an exclamation point. And all of the Fred numbers have been shifted by 33. So a quality score of 30 with a 33 bias would be a 63. And 63 is a question mark. So question mark really means a quality score of 30. Anyway, so this is the encoding for the quality score. Oops, I lost where we were. There we go. Let's get back to our read. So anyway, so this is our read. Actually, the beginning is some. Actually, there's two quality score encodings. There's the 33 shift and the 64 shift. Anyway, what do we got here? We got some capital Bs. Which shift are we dealing with in this one? Capital B. What's capital B? So that is a decimal 66. If the shift was 33, 66 minus 33 is a 33. So actually, capital B is a score of 33. Otherwise, in the 64 shift, it's a score of 2. It's not quite sure. Anyway, with enough examples, you can figure out what scale you got and so on. It's not really important to us right now, for everything. It's just reminding you what a Fastqc file looks like. Mainly, we're interested in the sequence here. So I'm going to copy that and put that in my clipboard. And I'm going to put that in a temporary text file, which you can't see that window, but I've got that saved in my clipboard. Now let's go and look and see the transformed file. That's the same file. And cutadapt, unless, basically cutadapt keeps the reads usually in the same order and so on, and it should be in this case. So if I look at this transformed file, actually on Hoffman2, you don't even need to do the gzip thing. On Hoffman2, the less is smart enough to know how to deal with gzip files directly. So I can just say a less in a gzip file, and it knows how to uncompress it on the file by itself. And here is the read. I could actually check that the read is the same name, but that read has the exact same name and metadata as the other one. I'm going to copy that line here and save that in my clipboard and click this. And that is so that I can use the without going through the pain of sharing another window here. Here, I'm going to paste in. That is the sequence, the original sequence. The Control-C there is just to get a new line here. I didn't want to run that as a command. I'm just kind of using this as a little display area here. That is the sequence from the trimmed file. And now we're going to align them. Find out where these align. Oh, there we go. That looks pretty good. And what bases are these? Okay, here's a little ruler. That's 10, 20, 30, 40, 50, 60, 70, 80, 81, 82, 83, 84. And now we need the tens digit. Sorry, the tens digit's going to be on the second line and not the first line. Okay. 5, 6, 7, 8, 84. Okay, where does it line up? That's, oh, do I have it? Oh, I got it. I lost a 9. There should be a 9 in here. Oh, my ruler's all messed up. Okay, the first line of the ruler is garbage. There we go. Oh, the whole ruler is garbage because the tens digit is also wrong. Okay, starting over. Okay, so there's, oh gosh, out of control. Okay, there we go. There's that line. I'm copying and pasting. There we go. That's lined up. Now I need a functional ruler. I'm still going to do the 1 digit first. Okay, there's this time with the digit 9 in there. Okay, there's 10, 20, 30, 40, 50, 60, 70, 80, 81, 82. Okay, now let's get the tens digit in, which is going to be on the second line. Okay, what bases are lined up? That is 11. The first aligned base is 11. The last aligned base is 74. So indeed, we have succeeded, and cut it up looks like it has cut out bases 11 to 74 as we intended. So that's a good spot check. It says, looks like things have gone right. So looks like everything is good. We are happy. We have completed the stage. We will now make a stage 3 directory, which is our cut it out output. I just made it. It's empty. We got to move our files. They're not in stage 2. They're in stage 1. Okay. It is everything with NT 11 to 74 Fastqc.gz, and you got to move into the stage 3. There we go. Now I'm kind of cleaned up. My stage 1 directory doesn't just have stage 1 again. My stage 3 directory now has my stage 3 output. And I've got this nice stage 2 to 3 script, which is literally the script I used to produce that. And so this is a reproducible analysis, and I have my breadcrumbs to figure out what I did later when I come back. And all is well and good. And now, how about on your own? You can this afternoon, why didn't you try a stage 4, rerun Fastqc on these new stage 3 files, and see if the problems that we saw in the Fastqc reports that we did before trimming are solved. They should be solved now, right? We should see flat things, and you know, the Q-scores should be more flat, and we should have this ACDT composition and wackiness and all this stuff. The adapter, you shouldn't have any adapter hits anymore. And so you should now be able to verify with a stage 4 rerun of Fastqc on the stage 3 output that the Fastqc reports look better now. Okay, so we've reached time. Day 2, we will start with bisulfite alignment with BS bolt. And then we will go as far as we can go from there with the pileup quantification and eventually the downstream analysis and statistics. And so on. So hopefully this is helpful, and you've been following along, and you've got the slides for reference, and you can play around in Hoffman, and we'll continue tomorrow. Okay, any questions? I can stick around for some minutes if you got questions. Okay, should I stop recording?
Yeah, sure.